uh, gain something. So today we are going directly to the presentations we have, and we have a cluster of presentations. And after that, we will take discussions and questions from the participants. Once again, on behalf of the organizing committee and the AfricaBP community, I'd like to welcome you to day 2 of the workshop. So to start, we are beginning with the first presentation, which is on high throughput meta barcoding, reference quality genomes, and annotation and Pan Genomes. This is going to be taken by Jacob Brandenburg and Jonas Kolach. So I'd like to, at this point, welcome to the podium Jakes to make his presentation on behalf of Jonas Kolach. Jakes, are you on the call? Can you please proceed with your presentation?
Yeah, you have to unmute him.
Yes, I have done that.
Great. Can everybody hear me and see my screen all right?
Yes.
Perfect. Give me 1 second here to get this set up. Okay, so yes, thank you for the introduction. My name is Jacob Brandenburg, and I'm the Global Marketing Manager here for Plant Animal Sciences at PacBio. Jonas had a last-minute conflict come up, but he helped me put this deck together, and he sends his best regards. So again, for today, I'm going to talk a lot about meta barcoding and also PacBio solutions for biodiversity research. Here's just a quick overview of what we're going to go through today. So I just want to start with some high-level applications areas in plant and animal sciences, and then we'll get into how PacBio is being used to characterize and monitor biodiversity. And as part of that, it's a good lead into how we generate high-quality reference assemblies using hi-fi reads, and then also expanding that into the different genetic variations that can be seen species-wide, and talk a little bit about Pan Genome assemblies. And so before we get started, here at PacBio, we break plant and animal sciences into 3 main categories. So we have agriculture, conservation, and ecology. For the focus of this talk, it's going to mostly be that conservation and ecology space. And we have an overarching theme of genomics in action. And so everything we try to do in the plant and animal space really fits into driving actionable insights from high quality data to really help drive change for a healthier planet. So if we take a look at the 3 main subsegments, starting with agriculture, the main types of customers we see in this space are from breeding programs, so people that are trying to use genetic information to gain better insight into breeding programs, pest and disease, and crop protection. And then also we're starting to see an influx of genome engineering customers really driving from the CRISPR-Cas9 takeoff in that space. On the conservation side, customers typically use this for capturing biodiversity, such as the African Biogenome Project and BGP. So a lot of these are larger biodiversity or consortia efforts that are multi-span, multi-year, and are really trying to capture the different genetic information for thousands of species relative to those areas. We're also used for species monitoring. And this is, again, happening very globally, and we'll talk about some more specific efforts going on in the species monitoring space. And then finally, researchers are using us for plant and animal biology to gain whatever insight they're looking for in that specific space. On the ecology side, we're used heavily for meta barcoding, such as Bioscan and the Barcode of Life, and I'll get a little bit more into these initiatives later on. And then also we have a pretty good metagenomics presence here with the plant animal community. And this is a look at the different biodiversity or consortia efforts that are going on globally. And PacBio is the main technology for all of these initiatives that you see here. So VGP, Earth Biogenome Project, ERGA, and of course the African Biogenome Project. And we've been a part of biodiversity consortia research for many years. So this timeline has obviously been updated to current, but this extends all the way back to about our technology's inception. So we can find articles and publications that we've been a part of all the way back to 2014 in the conservation space. And you can see here some of the different highlights that we have been a part of over the last years. And to get into some real world publications. So the first one I thought I would highlight for you today is Multi-Locus sequencing typing. And this was published in Fungal Genetics and Biology. And these researchers were using our next generation sequencing technology to do multi-locus typing. And they had their own proprietary analysis software that was used as part of this process. And the way that they were able to scale this to high throughput was by attaching a universal priming sequence onto the loci primers. And then from there, doing a second round of PCR to attach PacBio specific barcodes. And once the amplicons were barcoded, they were able to pool them together in massive parallel, and they were able to get to very high plex levels on a single run. And that is how they were able to be extremely cost effective with this type of sequencing. Another example here was sequencing different fungal populations from Botswana. And so what these researchers did was they had 3 different sample locations across Botswana where they sampled both environmental samples and also patient samples, because this fungal species can be breathed in and then cause different respiratory issues, but be present in a patient. And so if you look at the bottom here, you can see the map of this genome and sort of how it changes across the different environments and whether it was coming from the ecosystem or if it was coming from a patient. And they did a really nice job like mapping this genome as it changes across a different environment. Another great example of this is on the metabarcoding side. And so the biggest effort going on right now is the Barcode of Life. And so this is out of the University of Guelph in Canada, or Paul Herbert's group. And they did an initial pilot to see if they could use our instrumentation to replace all of the Sanger sequencing that they were doing. And so this pilot experiment was multiplexing 10,000 samples together on a single SQL system smart cell. And what they were able to find was that this produced much higher quality results than Sanger sequencing and was more cost effective than NGS and Sanger due to the plex levels they could achieve on our instrumentation. So you can kind of think of this as sequencing in massive parallel or like Sanger sequencing on steroids, essentially. And since this initial pilot experiment, they have ported this over to our new system, the Sequel II. And if you take a look at the heat map on the right here, they're actually able to pool up to 36,000 samples on a single run, where previously on different iterations of our instrumentation, they were only able to do about 10,000 amplicons. And some interesting data that's come out of this project. So this is a picture here of the testing site in yellow on the left. And this is Toronto, Canada again. On the right, you can see the different organisms that were found as part of this metabarcoding process. So what they've done is they've taken different environmental samples and they are sequencing them to see which species they can identify from those environmental samples. So the list on the right and the different colors all track back to different species. And some interesting ones that they found in Toronto was they found some instances of camel and they found some instances of elephant. And so they were a little bit perplexed at first as to what might be going on here, because why would insect and environmental DNA be containing camel and elephants, which are not native to this portion of the world? And then the reason that they figured out is the testing center was actually very close to the Toronto Zoo. And so they were able to have insect activity, sort of like feed on or interact with these different organisms, and that was able to make its way to the testing site. And so this is a really great example of how metabarcoding can be used to characterize species interactions. So you're going to have your viruses or your flies, etc., and how they interact with the host, and then also how the host is going to interact with other environmental samples such as nematodes. And so you can kind of see here that depending on where you get the sample from and at what time points you sample these different species from, you're going to be able to track that back to a different point in the life cycle for the different organisms involved as part of this ecosystem. And again, this effort out of University of Guelph is called Bioscan, or the Barcode of Life. And their main goal is to reveal species and the dynamic interactions that they have with like the host pathogen. And so this is a multi-year project with a lot of dollars, and there's a lot of researchers involved from a lot of different countries. And so shifting gears a little bit, now that we've talked about metabarcoding and some of the different biodiversity and consortia efforts going on globally, I wanted to spend some time talking about reference genome assembly, because this is also the technology that's really empowering a lot of these different efforts. And so to start with this, I thought I would highlight a multi-publication series that talks about how far sequencing has come over the last 20 years. And the main takeaway from these publications was that in terms of pretty much every assembly metric, when long-read sequencing came onto the market, it drastically improved contiguities, N50s, and a lot of other metrics. And you can see that in these three graphs on the right here, where the arrows are pointing to when long-read started to become adopted more widely by the community. And you can see that that has a drastic increase on the different metrics for these three graphs. And there are many reasons for this, but the first reason that we all know of is that plant and animal genomes are a lot more challenging than per se a human genome. And there are many reasons for this. So the first one is size and complexity. Plants often have very large genomes, and so to sequence that, it takes a different beast. And so long reads are just more naturally suited to be able to reassemble these genomes. These genomes also tend to have extremely repetitive content. So maize and wheat, for instance, were the two examples seen here. And with repetitive content also comes with very high GC content, which can be a challenge for alternative technologies. And then finally, each project is unique. So all these organisms are going to range in sizes, ploidies, and levels of heterozygosity. So often you have to come up with a custom strategy to extract, go through library preparation and sequence a majority of these organisms. And then even assemble them on the backside can also sometimes need a custom strategy. And so hi-fi reads that we generate here at PacBio are uniquely suited to tackle a lot of these challenges. And the reason for that is our reads are both long and accurate. And so because of the long read nature, we're able to span different transposable elements or structural variations that happen genome-wide. And because of the accuracy and the fact that we can track each read back to a specific molecule, when we go to do the assemblies, we're actually able to assemble each haplotype separately, as opposed to a collapsed assembly, which was the norm for many years of sequencing. And so again, because of this, PacBio is really the first technology that was able to break through that phasing barrier for a given species. And because of the different sizes and complexities of organisms, we also have to have different workflows. So we really break up 3 different workflows for different organisms. So on the left, we have our standard hi-fi sequencing workflow. This workflow has no genome size limitations. You just need to be able to generate 3 to 5 micrograms of material. In cases where that's not possible with smaller bodied organisms, we have our low DNA input sequencing workflow. This one is really designed for species that have genome sizes up to about 1 gigabase, and you need about 300 nanograms of high quality, high input starting material. And then finally, and the one on the right is a little bit newer to us, we have our ultra low DNA input sequencing. This workflow does use long read amplification, or excuse me, long range amplification. And you have about 5 to 20 nanograms of starting material that's needed. And we're really targeting genome sizes of about 500 megabase pairs or lower. So this is for those very microscopic, tiny organisms that may not even fit into the low DNA sequencing workflow. We've also recently purchased Circulomics, which is a DNA extraction company. And so they are now under the PacBio umbrella. And the reason for this is we know that to have success from a lot of these projects, it starts at the very front end. So if you don't have high quality, high molecular weight going into our library prep procedures, you're not going to get high quality data coming off of the back end. And so we felt that Circulomics had the best product on the market for extracting high molecular weight DNA using their nanobind disk technology. And so we're excited now to be able to offer those kits as part of our overall workflow. So now you can have PacBio reps support you from DNA extraction all the way through DNA data analysis. And now I want to walk through a couple examples of these workflows. So the first one is going to be an example of our ultra-low DNA input kit. And this was really coming from a human face mite out of the Cal Academy of Sciences. And so face mites are something that everybody has. They are an unculturable eukaryote, and we tend to inherit them from our parents. So previous attempts at sequencing this genome were unsuccessful. Very few percentages of the reads were able to map back to the face mite genome. And due to this low read coverage, they were not able to assemble the genome. It kind of fell apart at the assembly stages. So once this ultra-low kit came out, these researchers got excited to try this experiment on that workflow. And they were able to generate about 40% of the reads to face mites, where the other 60% tend to be coming from the host. They were able to assemble it into about a 52 megabase genome, and it had a really good BOOSCO score to different arachnid genes that were in the database. So they were extremely happy with this assembly. And kind of going on the other side, this one was a locus assembly done by the USDA here in the USA. And we were actually sampled some of these locuses from Africa. And so they were pleased to announce earlier this year that they have a really good reference quality assembly for the voracious desert locust, which causes billions of dollars of agricultural damage every year. So a very invasive species and a very highly studied species. And the USDA has made this public on the NCBI database, and they were able to assemble this into 8.8 gigabases, and they were able to get all 12 chromosomes fully assembled. And so the authors went on to say that compared to the fruit fly, sequencing the desert locust is kind of the equivalent of comparing an 18-wheeler to a compact car. So they were very happy to be able to tackle this very large insect genome. And as we study these genomes and put together these larger biodiversity efforts, what we come to realize is that having a single reference for a species does not encompass all of the genetic invariation that can be included across those species. And so what we are coming to realize as a science, or as a sequencing community, is that we need to generate pan genomes for species and not just single references. So a pan genome is going to be a compilation of a bunch of different assemblies for a genetically diverse population, making sure that we're sampling species across that entire genetic diversity. And what you do is when you start adding those assemblies together, you start to have this picture come into focus that we call the pan genome. So the pan genome is going to have core genes that make up that species, and core genes can be tracked back to critical functions. And these genes are going to be very slow to evolve over generations. And you're also going to be able to see dispensable genes. And so these dispensable genes are really what house those adaptive traits and what make a lot of these species able to adapt to changing environments. And invasive species are also very good at adapting. And so a lot of those traits can be tracked back to these dispensable genes. And then the remainder of the pan genome is going to be made up of unique genes. And unique genes are going to be specific to an individual or map back to a very small percentage of individuals in that species. And so this can also be informative if you start noticing that a specific individual is thriving better in a specific environment, that may be a good indication that they have some genetic variation going on genome-wide that is allowing them to thrive better in that environment. And so why is this important? Again, pangenomes are really going to be able to help exploit genetic diversity of wild relatives, and that's really going to facilitate our understanding of agrogenomics traits, as well as assist breeding strategies as we move forward. And this is especially important in plants, because as humans, we are all about 99% genetically similar from one individual to the next. So there's not a lot of genetic differences. This is not the case in plants. If we look at maize, for example, maize is only going to share about 50% of its genetic makeup. So if we're only looking at one individual, we're not accounting for all of the genetic diversity and structural variations that are going to exist species-wide. And here are some examples of that in the biodiversity space. And so this was a great publication done on the Mediterranean mussel, which is a very invasive species here in the US. So they did a pan-genome approach to really understand the different genes involved to stress response and survival for this species. And this pan-genome really laid the architecture to understanding those adaptive roles and those traits species-wide. And another great example out of the USDA, again, was with the Asian giant hornet. And this species became very popular right around the time that COVID-19 was being introduced here in the Americas. So we found this invasive species for the first time ever. And there was a huge concern because this species is known to attack like honeybee hives, essentially, and that would kind of have broader implications on the larger ecosystem. And so once this species was found, very quickly generated a pan-genome, and they were able to use that reference to understand how these species might adapt and how we can combat them. And we were able to address this species before it was able to become established. And then finally, the last piece of this pan-genome puzzle is genome annotation. And so a lot of times with these reference assemblies, you have to have them properly annotated, and that really helps the pan-genome picture come into focus. And so the assembly that I thought I would pull out here for you is actually the ANT, so a true annotation. And they used isoseq, which is what we call our RNA sequencing method here at PacBio. And so what our isoseq method does is it sequences full-length isoform and full-length cDNA. So it makes it the best technology for annotating genomes. And when they paired isoseq to this genome of interest, they were actually able to correct different gene models from both the brain and the fat body and ovary systems. And using those updated models, they were able to get increased mapping from their short read data by about 44%. So this is a great example where long reads and short reads can play together as one technology to really drive forward the different questions that researchers might need to answer. And with that, I will just leave you with PacBio reads are currently the most complete and accurate long read sequencing technology on the market. And you can see on the right here the different applications that we enable. And the one I did want to give a call out to was epigenetics. So this year, just a couple months ago, we launched 5MC calling as part of our assembly process. And so now if you're using hi-fi reads to do an assembly, you can also get 5MC calling for free. So these are just different calls that are stored in the sequencing files that you can visualize later if interested. And so we're hoping that this new level of information is going to be able to really drive biodiversity and consortia efforts forward into the next 20 years of sequencing. And with that, I want to thank you for your time today, and thank you for inviting us to speak for part of this, consortia today. Thank you.
Yeah, thank you so much, Jake, for your presentation. We hope you'll still be on the call. We will take the second presentation, and thereafter we'll proceed for discussion. And then you can address questions that concerns you, as well as for the other second presentation altogether. Thank you once again for your presentation. at this point, I would welcome to the floor the second presenter for this section, and he is going to be talking on apply different methods to resolve complex plant genomes. I believe this is going to be very interesting. I now welcome Xin Liu from NGI Technology, China, to give his presentation. The floor is yours now, Jay. So, to mute you.
Yeah, okay. I think you can hear me now, and I will share my screen and start my presentation today. So it's great honor to be here talking about how to apply different kind of method to resolve different kinds of plant genomes. So I'm Xin Liu from BGI, and I am a plant research scientist at BGI. So I will talk about the plant genomes that we have assembled and analyzed. So first of all, I think I will give you several examples, starting with the foxtail millet genome. That's the genome we sequenced quite some time ago. I think it was published in 2012. So 10 years ago, we published this genome. This grass genome with relatively small genome size. And this is just like the process that we usually use for all different plant genomes. I mean, in previous times, I mean, when you do not have like a very effective long range to be used. So the first thing about the genome, to start the genome research is to get the information about the genome, especially the genome size, the repeat content, like the previous speaker mentioned about. There are different aspects of the genome which might affect the sequencing and the assembly, especially about the genome size, the repeat content, and for plants, specifically that the heterozygosity. So those kind of aspects can be obtained from the different kinds of analysis, including like the K-mer analysis. So for the foxtail millet genome, we got this K-mer distribution shown here, indicating majority of this genome to be relatively easy without much high repeat content. So if you have higher repeat content, you can have a secondary peak here. If you have a repeat content, that would result in the secondary peak here. And if the high heterozygosity, you have the secondary peak here. So from this K-mer distribution, we think this genome is relatively easy for us to sequence and assemble because of the genome size, 500 megabytes, about that size, and the heterozygosity should be low, and the repeat content should also be low. So what we did back to them was to apply the second generation short reads, different insert size libraries to do the sequencing. And this is the workflow of the software to use the short reads to do the genome assembly. So first of all, you'll get the debrugging graph, and then resolve the graph to get the quantic assembly that the sequences without assembled sequences without any gaps there. And then because you have longer range kind of linked rates, you can then use that information to link the contigs further to get the scaffolds. So usually you get the scaffold assembly. This is the assembly statistics we have back to them. As you can see, especially from the contig M50, scaffold M50, those kind of statistics reflect the continuity of the genome assembly. And back to them, we do not have other technology like the high-C effectively available back to them. So what we did is to resequence one individual and to identify some genomic variations and apply these variations, genotype those variations in the population, in the inbreed breeding population, and construct a genetic map. So using this genetic map, we finally anchor the scaffolds to the chromosomes. So this is what we did for that genome, which we published in 2012, how we do the sequencing, how we anchor the chromosomes, and how we further analyze the genome. I will not go to details about the analysis today because we will have several different examples about the genome assembly. But here just indicates what kinds of analysis you can do based on the genome. So you can get some preliminary information like the repeat, like the genes, where are the genes, what kind of gene families are there for this genome. And you can compare this genome to other species to analyze the evolution of the species, whether, especially for plant, the whole genome duplication events are there for this species. And you can compare this genome to other related species in chromosome level to give you this kind of chromosome rearrangement analysis result. So those are some kind of analysis you can do. You can also further look at some specific gene families, trying to relate that with the evolutionary process of the species. Then that is just a simple genome. That's just a small genome. Then we have the second example, that is the Ginkgo genome. This is the genomes per species. Quite well-known species. So the genome size of this species is actually 10 gigabyte, around 10 gigabyte. But if you look at the K-mer analysis, what you can see here, although the genome is relatively big, I mean 10 gigabyte, it's quite clear that the major peak is here, and some secondary peaks like here indicates some repeat content, and maybe here also further indicate repeat content. So repetitive sequence is one problem of this genome, but actually from this kind of K-mer distribution, we think we can do it even back to them when we only have the short read data for this species. So this is what we can do. We can achieve using just the short reads. You can see the M50 size scaffold and counting M50 not so long, but we can get one longer than 1 megabyte of the scaffold M50, even just with the short reads. So we assemble this genome and release this genome several years ago, and later some other groups, they're using the new technology, the long reads, also to assemble this genome. But from this K-mer analysis for this Ginkgo genome, although the genome size is huge, we can see it's relatively easy for us to sequence and assemble. And for Ginkgo, we also carry out further population resequencing. These are the samples we collected worldwide. We sequenced more than 500 different individuals. We resequenced them at around 5 times of the genome size, 50 gigabyte of the data for each individual. And then we used Hi-C here to anchor the previously assembled genome to chromosomes. So we get a chromosome level assembly. And along with the population analysis, we carry out some very easy kind of very straightforward kind of analysis to associate the sex determination with the genome variations. What we found is a peak here indicating very strong association between the sex determination and the genomic variations here located on chromosome 2. And then we further looked at details of this region to finally locate around 5 megabytes of this region to be the sex determination region of this genome. But actually, we need better assembly at this region, at least in this region. So we further use long reads to further assemble this region because, as you know, this sex determination region, they might contain high content of the repeats. So the assembly of this region would be quite difficult. We are still working on that, but this is just some example in which we can use a simple strategy to assemble this kind of large genome, but looking at details, we might need to apply further kind of long reads to get more information. So about the long reads, we are also applying to other genome species. For example, this is the one we recently published in Nature Plants, the cycas genome. As you can see, for this cycas species, we analyzed the genome content again using the K-mer analysis. As you can see here, comparing to the Ginkgo one, so the K-mer analysis indicated relatively difficult genome for us to sequence and assemble. So the major peak seems not to be very clear, and you can see very notable kind of repeat content here. I think from our analysis, we found that for Ginkgo genome, the repeats, they are long-term, they have evolved for a long time. So from copy to copy, they might be different from each other. This is why we think we finally call them the low copy repeat content in those genome. So for the cycas, we use long reads, nanopore, and high C to get the genome assembly. As you can see, we can get way much better, 12.1 megabyte of the Contic M50. And the final genome assembly also around 10 gigabytes in 11 chromosomes, because high C can be used to anchor them to chromosomes. The other example is about the lychee genome. Also, we participated in. It took us quite a long time. I think longer than 7 years to finish the genome, actually to publish this genome in Nature Genetics. So the feature we found is comparing to the genomes, is we get very high heterozygosity for this species. So as you can see, we estimate the heterozygosity to be 2.27%. This is quite high heterozygosity. But the genome size is not that huge, around 470 megabytes of the genome size. When we use short reads to do the genome assembly, we assembled around 800 megabytes, indicating that we can, because of its very high heterozygosity, even using the short reads, we can assemble both haploids there in this genome. So the lychee genome, the other example about the heterozygosity. So but for plant genome, heterozygosity is definitely the other information you need to consider in addition to the genome size. Then the examples about the Begonian genomes we recently published. So this is the strategy we used to assemble those genomes. We used both the linked reads and also the synthetic long reads, or to say the linked reads, and also the long reads for some genomes. As you can see, we sequenced more than 70 different Begonian species, and among them, we selected 4 of them to be further sequenced to get the genome assembled. From the K-mer analysis, we have the 4 K-mer analysis indicated here. As you can see, we get clear peak of the first and the last genome, especially the last genome. So this kind of k-mer distribution just indicates the genome content to be relatively simple. We can easily assemble this genome, but for the other two, as you can see here, for some k-mer lengths, you just cannot observe, you cannot know which is the major peak. You cannot find the major peak here for this one and also for this one, indicating either heterozygosity, repeat content, they are affecting the genome, and there should be high amount of the repeats or the high heterozygosity. That would affect the genome assembly. So what we did is to use the synthetic long rates to assemble the first and the last genome, and to use the long rates to assemble the complex genomes, the in-between two complex genomes. So what we can get, we have a table to show you about applying different kind of methods to assemble this 4 genomes. So first of all, we use the synthetic long rates, either the 10x linked rates or the single tube long fragment rate, SDFR we have. So we use both of the synthetic long rate method to sequence all 4 genomes. And as you can see here, to show you some of the assembly statistics, especially the N50 size, as you can see, N50 size here, relatively good for the first one and the fourth one, but very low N50 size for the second and the third one. This is consistent with what we anticipate from the k-mer analysis, right? So we think we can just use the synthetic long rates for the genome assembly of the first and last one, but then for the second and third species, we add long rates here. As you can see, it helps us to improve the genome assembly from the previously just using the synthetic long rates from some around 100 kilobases to 500, around 500 kilobases, those kind of N50 size. This is what we can get. Also, one further information to mention about the genome size. For the last one, the genome size is relatively smaller, like 400 something megabytes. But the previous 3, they have a larger genome size, like 700 megabytes or so. So the Begonian genome, again, indicating the different kind of strategies, different kind of sequencing data you can use to get the genome to be assembled. So especially for the Begonian genomes, this is the one we recently published, because we sequenced more than 70 species, and many of them, 4 of them, we want to assemble them to be better kind of assembly. So we use different strategies. What we did is to know the genome complexity, trying to know better about the genome complexity, and then to adapt the suitable, the better kind of strategy to assemble those genomes. So those are the examples of the plant genomes, but we have more of them. We have many of the plant genomes to be sequenced and assembled in our team. And back to 2017, we initiated this 10KP that is aimed to sequence the complete genomes from more than 10,000 plant species. We have the plan to sequence seed plant, non-seed plant, and also the green algae, and the other related species, as you can see, what we plan to do. And the strategies, the methods we're trying to use is indicated within this workflow. So first of all, we would like to know the information about the genome, the complexity of the genome. We have the examples showing you that no matter the huge genome or the high heterozygosity. They might not be. They are the major issue affecting the genome assembly, but they might not affect the one you are assemble, right? Just like the Ginkgo and the lychee genome. Actually, you can use the simple kind of way to assemble the genome. But if the genome is not that easy for us to sequence, we actually need to use like the long rates along with the high C, which can help us with the chromosome assembly, and also some transcriptome data to help the gene annotation, because we need to get the genome information. That would be the major information we want to release for those kind of genomes. So these are the strategies, methods we adapted. And for the plant genomes, there are many kind of research questions we would like to explore. Those are some of them that we focused on, listed here, because from different clade, for different clade of the plant species, there are different kinds of SAMs. Like for some algae species, we can look into the arranging of nitrogen fixation, and also for green algae, we can look into like how the green plants, how the later kind of plants, they can go to the land, the terrestrialization, that would be the focus. And also for flowering plants, how the flower related phenotypes, they evolve, how they evolve, those kind of scientific questions. We have examples of like the algae genomes to be published, like those two examples showing about different algae genomes. We analyzed further to resolve some of the problems we have mentioned in the previous slide. And again, some more algae genomes example. And bryophytes, the other same, because they are very small, and you might not get enough amount of the tissue for DNA extraction. So although the genome size is not very huge, 450 megabyte in those kind of average. And we have examples, we have some data released of this bryophyte genomes are listed here. And for genome spur, basal angiosperm species, I think we have shown you about the cycas, Ginkgo genomes, and we have more to be sequenced right now. And for flowering plant, we would like to sequence more to construct a family level kind of species tree. For family level, we have representative species to cover all the different families, also under progress. But I think there will be one problem, if not so many, but one of the major problems we encountered is the sample. So you need to get the sample. It's not for the crop species which you can get enough sample, right? You can get quite a lot of sample for one individual for the crop species, but for some species, just like the bryophytes, we do not get enough sample. And you need also to think about the sample shipment. You get the fresh sample, you ship them, it becomes just not that fresh, considering about the logistic problems. And also the DNA extraction, you need to get the long fragment DNA. Also, the last speaker mentioned about some methods they developed. So we're also trying to solve this kind of problem, how the sample can be prepared, shipped, and also the DNA extraction can be more effective. We have some internal kind of protocol that we also share around with our collaborators. So the last slide, I think I applied, I submitted the abstract indicating some more technologies, including like the example shown you here, especially for the plant that do not have much kind of background research on. the example showing you about the peanut genome study, we established the gene reference genome, then we carried out the population resequencing. And recently, we also used our stereo-seq technology, that is the in situ capture method to capture the gene expression on the slides. So you have the sections, and you put the sections onto the slides capture array, and it will capture the messaging RNA. And after sequencing, you know which messaging RNA comes from which part, which location. And finally, you will get this kind of information, like where on the root are expressing different kind of genes. I don't have time to go to details, but I just mentioned this a little bit. I think if later we have more time, we can share this around that applying the new technologies, new sequencing technologies, we can have more information, especially for those species without previous in-depth kind of researches. So that is all for my talk. I think around 20 minutes. I would like to thank all the collaborators. These are the major collaborators in the 10KB project, and also the publications we mentioned in this slide. Thank you very much. I would like to take questions if you have. Thank you.
Yeah, thank you, Zane, for the presentation. And awesome, thank you. So at this point, we are going to the next part of this section, which is going to be a panel discussion. This is the time to ask questions orally, and then the questions that were posted on the chat box will be taken as well. And if there are any other thing that you want to ask, then this is the time for it. First, I would like to know if there are people who would like to ask questions. You can indicate with your hand raised, so we can ask you to ask your question. So as we're doing that, we will also take from the chat box for the presenters to answer. So does anyone have a question? Now we have Jacob on the call, and we have Zane on the call to answer questions. Okay, I will give the floor to Tim. Tim, please go ahead and answer your question. And
hi. Yes, I'm assuming you can hear me. Hi, this question is for Jacob from PacBio. First, I'm just going to admit upfront, I don't know much about the PacBio technology other than it's one of the two major long read technologies out there. I quite enjoyed your presentation, but my question for you in the context we have here in Africa and Southern Africa is, what is the relative cost of using a PacBio system for sequencing compared to Illumina short read sequencing?
I think, thank you, Tim. Let me just also add a follow-up to that so that Jacob can answer both of them. Someone also wanted to know the difference between the PacBio and the nanopore systems and the various reads that it generates. So. I think Jacob needs to be, Jacob, could you please unmute?
Thank you. Perfect. So yeah, great question. Thank you, Tim, for asking. So in terms of cost, I would say that short read does tend to come in a little bit under, both right now on the instrumentation and the data generation side. We can get around some of those costs by the amount of coverage needed. So with short reads, you tend to need very high levels of coverage to do whole genome sequencing projects. With PacBio, we recommend about 15 to 20x coverage per haplotype. And so due to like the lower coverage amounts, we can get around that. And then I would say today we're also very flexible on pricing. So, you know, I feel like if you do have pricing needs or want to find PacBio projects, we can help connect you with cores and other institutions that can do the work for you, or if you need to bring it in-house, I know that our team is more than willing to work with customers on that front. In terms of the differences between the two technologies, so you're correct, like Nanopore and PacBio are the two players in the long-read space. I would say the biggest difference comes down to the data quality and the data type. So the way that we generate the data is with an enzyme. So we use a polymerase, a sequencing polymerase, as the engine to really capture the information, where Nanopore is going to use a pore that they kind of feed the DNA through. So the differences between those two is that our error rates are more random and ONT's error rates are more systematic. And so the way that we make our libraries is we ligate on circularized adapters on the ends of our DNA fragments. And that allows our polymerase to make multiple passes around a single molecule. And because of the random error rates, we can collapse the multiple reads from a single molecule down together and come up with a very highly accurate read, where ONT does not have the ability to read the same molecule multiple times at this point in time. And so their raw error rates tend to be lower. And so I'd say, to me, that's the biggest difference between the two. ONT does, because of the pore, they can still get to very large fragment lengths. So sometimes you can see HiFi and ONT being used together in terms of ONT reads can scaffold HiFi reads, because HiFi read lengths right now are capped at about 30 KB is probably the largest HiFi reads that you'll see. So yeah, thank you, great question.
Yeah, so Taiwo, if you can permit me just to follow up with that. So what, and also a follow up to Tim's question, what is PacBio, I mean, how are there efforts on the way to increase the PacBio systems, to increase the footprints, basically, because I think at the moment it's a bit sparse, I would say, on the continent.
Yes, so again, there can always be some challenges depending on where a company is based and how you can get into different countries. But I think that as the plant and animal community, we recognize the importance of Africa as a continent that is going to be able to help address and solve a lot of the climate change and conservation problems that we're having right now globally. And so I think that at like every ag conference I've been to this year, everybody talks about Africa as the means to really help address a lot of those problems. So I think that at least at PacBio at a very high level, we recognize the importance of that and are definitely thinking about ways to try to help penetrate and make the technology more accessible worldwide. And so we have definitely placed a couple boxes in Africa as of now, and we're always very willing to work with customers or people that are interested. So I'm happy to leave my contact information after the chat. And if people want to have more discussions on that front, we're definitely open to and willing to have those conversations and how we can further research and development here.
Yeah, thank you for providing answers to that questions. Yeah, so does anyone have a question to ask? You can indicate, but while this is ongoing, we have more questions in the chat box, which I'd like to read out. And we have a question that is the software for the Genome Assembly open source? So this is one of our participants who was interested to know about this. And I believe that you can answer these questions for the participants again.
Yeah, I think this would be to Shin, do you?
Yeah, this will be to Zane, yeah. Please.
Yeah, we use different, for the examples I mentioned, we use different kinds of software there. So I think most of them, they are freely available. So you can use them for your project. That would be okay. speaking of whether they are open source, I think majority of them should be, because they are this kind of bioinformatic software pipelines developed by researchers. Many of them should be open sourced, but you can freely use them for your project. That's the case. The same is that I think based on your project, especially about the genome content, the genome complexity of your species, and also the data quality you have. You might want to choose, you might want to select the right one for your assembly. So that's the major thing you want to consider. I think that's the answer for that question.
Yeah, thank you very much. We still have more questions for Zane. Here, there's a question on what is the progress of the 10kp in terms of genome sequencing? And there are also other follow-up questions which I would like you to take together. What is the best assembler to use for autaploidy genomes? And then there is this question on, do you have BGI service laboratories in Africa? If yes, how can African community benefit from their services? I believe you can take all of this together.
Yeah. Okay, thank you. So for the first one, I need to find that.
I can remind you, it's on what is the progress of the 10kp.
Oh, okay. Thank you. So for the first question, I think we have the sequenced. We have attempted samples around 2,000 species. Just like I have mentioned, the sample shipment is one of the major problem we are encountering for now, because usually what we encounter is our collaborator, they prepare the samples for the biodiversity researchers. So you might not get very fresh sample, even you are the sample collector, right? So when they get the sample, they think they are fresh, but when we get the sample after the shipment, the condition might be different. So this is the same. We received around 2,000 different samples. For majority of them, we carried out the genome survey, that is to sequence some short read data to get more information on this genome, because again, this is different from the crops. Not so much information, previous information available. So the genome survey would be important for us to understand the genome complexity. So I think majority of them, more than 1,000 species, we get the short read data. And independently, we also have the other project to sequence just the short reads. We sequence more than 3,000 species within a botanical garden in China, from China. So these are the sample amount and also the short read sequence. But for the final assembly, I mean, to get the genome to be actually assembled to relatively good continuity, I think we get around 500 genomes already assembled, but we will release the data later on. I mean, recently we will release the first bunch of the genomes we have recently assembled, in addition to the ones we have already published. So these are the progress. I think later on we will also, we are collaborating with AfricaBP, and if there are some chance, we can further share about the recent progress of 10kp if we have more time. Then this is about the first question. Second question.
Yeah, the second question is, what's the best assembler to use for autaploidy genomes?
Okay, thank you. For the autoploid problem, they are similar. I think first of all, you need to know what kind of sequencing technology you are applying. Majorly, it's related to that. I mean, if you use short reads, you use synthetic long reads, you use the PacBio or the nanopore long reads, you definitely have some software suggested for you, no matter it's it's polyploid species or just haploid, right? So the first SEM you consider about the software, that is the data you have, and secondly, I think there are some software available, although we do not use that much, specifically designed, adjusted for different autoploidy species maybe, but we are not using that much. So I do not have that much experience on that. I will look into some of our examples. If we have, maybe we can have more information to share after that. Okay, that about the second one. The third one, whether we have the service to be provided locally in Africa, I think so. We have the sequencers there, at least in several countries in Africa. The one we recently collaborating with is in South Africa. The team there is right now helping us to at least to try some plant genomes to be sequenced and maybe later assembled locally there. We will release more information on that site, either the BGI Genomics or the AMGI. Our two companies there, they will release those kind of information in detailed information on that.
Thank you so much, Zane, for your answers.
Thank you.
Yeah, but I'd like to ask Jacob if he can throw a little bit of light into to answer this question from one of our participants. He's interested in knowing what are the procedure or software for assembly and analysis of genomic and transcriptomics data. So could you answer this?
Yeah, so we have a free software called SmartLink that is open source and free to download off of our website. And that's a really good starting place to do a lot of different assemblies and other pipelines. And if you need to do something that is a bit more command line or have a bit more flexibility than what can be offered in SmartLink, we can definitely connect you with like a Bioinformatics FAS or Bioinformatics services at PacBio to kind of walk you through what might be third-party softwares that will work better for your specific process. But I think like Jin kind of alluded to, that most things are open source these days, and it's just kind of finding the right fit for the organism that you're working in.
Thank you very much, Jacob, for the answers to that question. Now we'll have one hand raised, and we'd like to call on Sunday to ask his question. Sunday, please unmute yourself and ask your question.
Okay, can you hear me, please?
Yes, please go ahead.
Okay, thank you, presenters, for those presentation. But I'm actually interested in the Pan Genome. And I actually saw in one of the slides a Venn diagram that shows the dispensable gene, the core genes, and the unique genes. And what I just want to know more is the Venn diagram, is it related to using the Pan Genome, which comprises those unique genes, in comparing a particular species of or genome of a particular organism, or across more than two organisms? I would like our presenter to share light on how it can be used for comparative genomics.
I'm happy to take this one first pass. So I would say that typically when we talk about a Pan Genome, at least at PacBio, we're talking about within one species. And I didn't shed a lot of light into potentially like number of organisms needed. And so it seems like the number that we tend to recommend people start with when generating a pan genome is 20 to 30 different individuals from one species. And just make sure that of those 20 to 30 individuals that you have a pretty diverse subset that you're sampling from. So, you know, not all one species from a specific area or a specific type. And then for those species, you're going to want to have the reference genome information, the whole genome sequencing information, and then also complete with annotation.
Yeah, thank you, Jacob, for the answer. We want to ask if there are still more questions people are interested to ask. You can indicate, and then you can ask your questions. More questions? Okay, we have one coming, which is for the Pan Genome approach. What kind of coverage would you recommend for each of these 20 to 30 specimens or species?
Yeah, great question. So
yeah.
Again, our typical recommendations for the plant side is, I would say, you know, let's just say 15x coverage per haplotype. So that's the recommendations in terms of coverage. So it's going to definitely depend on the ploidy and complexity of your organism. On the annotation side, you can get away with probably doing 1 or 2 of those species on a single SMRT cell for the isoseq method for the annotation. So however many cells you would need to get to that 15x coverage per haplotype, and then also maybe additional half cell or cell for the isoseq portion.
Yeah, thank you, Jacob, for the answers to the question. So does anyone still have questions to ask Zane and Jacob based on their presentations? Okay, so you can always send in your questions. I'd like to know if there are questions on the channels, those viewing from the channels. You don't? YouTube channels and other handles?
I can't say none at the moment. None at the moment.
Okay. Great, thank you. So on this note, I'd like to thank very especially our presenter today, Zane Liu and Jacob Brandesburg, for their time to give a talk on this platform. We really appreciate you. So, and we bring this section to a close, and when we have 10 minutes break to come back for the second section for today. So thank you and enjoy the break.
So Taiwo, 10 minutes,
Yeah.
maybe what time?
That will be to be
3:25 GMT plus.
Yeah, 3:25. We're back.
GMT plus 1.
Yes, GMT plus 1. We'll be back at that time. Please endeavor to return. Thank you. Hello, everyone. Once again, I'd like to welcome you back to this second section of the workshop. So in this section, we are going to be taking 3 presentations, and thereafter we'll have the panel discussion as we did for the first section. So the first presentation is going to be on what is in my genome, an introduction to Bloop Toolkit. And this is going to be taken by Sujay Kumar from the Wellcome Sanger Institute, UK. After this presentation, we have the next presentation on the Uniprot database. This is going to be taken by Nigi Tiangi and Ketwana from the Universal Protein Resource, UK. And then after their presentation, we'll have Diplomics, a South African national research infrastructure program supporting African biodiversity. This is going to be taken by Tim Nema and Shane Murray and Peter Moyo. And after all of this presentation, we'll take a panel discussion, which is going to be participants asking questions, directed questions to the presenter. So to start this section, I will call to the floor Sujay to give his presentation. Please unmute yourself and begin your presentation, please.
Okay, now I'm allowed to unmute. Hello, everyone. I'll just share my screen again. Okay, just a second. That's my outline. Here we go. So hopefully you can all see my screen. Just say yes or no.
Yes.
Fabulous. So my name is Sujay Kumar. Thank you very much for inviting me, Africa BioGenome. I'm very happy to be here. My talk is called What's in My Genome? An Introduction to Blob Toolkit. And as Taiwo said, my name is Sujay Kumar. I'm a senior bioinformatician at the Daventry of Life project at the Wellcome Sanger Institute. I know Taiwo said he would make all the presentations available later, but in case you want it now, it's tinyurl.com/abp-btk. It's on the screen. But you can follow along and you can get the presentation link later. So, what is the Daventry of Life? I said I'm at the Daventry of Life. Well, it's a project just like the African Biogenome Project. And both are part of this global initiative called the Earth Biogenome Project, where we're trying to sequence all 2 million eukaryotic species. It's a huge effort, lots of people around the world. So African Biogenome Project is trying to sequence 100,000 species in Africa, whereas we in the UK are trying to do this Daventry of Life project, where we are trying to sequence the genomes of 70,000 species in Britain and Ireland. And my role is to develop and promote bioinformatics tools, like the one I'm going to be talking about today, for the Daventry of Life, but for everybody, because the projects that we make are all open source, they are applicable to everybody, so they are useful to the African Biogenome Project and to all other Earth Biogenome projects. So how do you sequence a genome? So already today we've had 2 talks which talk about this general process. On the simplest level, you collect the sample, you extract the DNA, you sequence the DNA. Here I've shown PacBio, but you could use Illumina, you could use Nanopore. And then whatever sequences you get, you have to assemble them together. Sounds straightforward. Obviously, each of these steps is complicated, but let's assume you've done everything properly. What can go wrong? One of the things is that other species may be present in your sample, in your assembly. And the reason for that is it could be a contaminant. So it could be a lab contaminant or an air contaminant or even a human contaminant, or it could be a parasite of that organism that you're trying to sequence, or it could be the gut microbes and bacteria that live inside the stomach of that organism or on the skin, or it could be a fungal or viral or bacterial infection. Or it could even be symbionts, so things that always come along with the organism. For example, there is a fish which has bacteria that live in a little sac inside it, which is what causes it to have bioluminescence. So those symbionts, whenever you try to take that fish and you try to take a sample from its innards, you might get some of those bacteria as well. And lastly, you might even get endosymbionts, which is even more complicated. Some insects, the Wolbachia bacteria don't just live inside the body, they actually live inside the cell of the eukaryotic species. So you can have different levels of organisms that come along with your sample. So you have to be careful about this. And so rather than calling them contaminants, I'm going to call them cobionts, because cobiont just means other organisms that come along with your organism. Why is this important? Here's a great example that I was involved with. So in 2015, this paper was presented in PNAS, and it made a big splash, evidence for extensive horizontal gene transfer for a tardigrade. Now, tardigrades are very small creatures. And when these people sequenced the tardigrade genome, they didn't realize they had accidentally sequenced all the bacteria that live outside it. These were not bacteria in the genome. These were bacteria outside the genome. So within 2 weeks, we wrote a reply on bioRxiv, and within a month, in January 2016, we had posted a reply on PNAS, and we showed the opposite. No evidence for extensive horizontal gene transfer. So you might make a big high-profile claim without realizing that your findings are based on a contaminant rather than on the actual genome of the organism. So you have to be careful. This is why it's important. The main question I'm answering today, as you saw from my slide title, is what's in my genome? And the answer to that, I'm going to suggest to you when you sequence a genome is use Blob Toolkit. So what is Blob Toolkit? Well, a Blob Toolkit, the first thing I have to tell you is it's a toolkit that allows you to analyze blob plots. And of course, the next question is, what is a blob plot? So I'm going to show you a blob plot on this screen, and I'm going to build it up one by one. So this blob plot is for the Genome Assembly of Didymella aricidicola. I hope I said that right. What is it? It's a plant pathogen fungus. So it's a eukaryote, it's a fungus, and it causes the leaf blotch of peanuts, like you can see, and it's found in Africa, in Asia, I think also in America, but slightly different species. So here's what a simplified blob plot looks like. And you might think, what's going on over here? Well, very simply, you're seeing two blobs, a blue one and a green one. And now I'll explain what they mean and what the axes mean. So each circle is a contig. And a contig means it's a bit of assembled genome sequence. So you've done your genome assembly and you've put it in the Blob Toolkit software. And so each circle is a contig. So the size of the circle is proportional to the size of the contig. So you see these very small circles, they're very small contigs. You see this bigger circle, that's a bigger contig. And you see this large one, that's an even bigger contig. This picture does not have the scale, but just get that idea. The x-axis tells you the GC content of each contig. So if a contig was only 10 nucleotides long, obviously these are much longer, then if you count all the G's and C's, here there are 6, and you divide it by the total length, which is 10, you get a GC content of 0.6 or 60%. So in this case, for this contig, for this circle, the GC content is 0.53 or 53%. And on the y-axis, we have what's called the sequencing depth or the coverage of each contig. And by coverage or depth, what we mean is if you map the reads that went into the assembly back to the contig. So this is the assembled contig, this green line, and the black lines are the individual reads. In this toy example, you can see that the average depth is around 5 or a little under 5. So over here, when I take the y-axis for that same blue circle over here, we move it, and this is a log scale, which you can see from how the numbers are changing. I can tell you the coverage is 16.5. So we've plotted the contig. We already know the size. We know the x-axis is GC. The y-axis is coverage. And the last thing on this plot is color. So the color is a taxonomic assignment based on some tool that we can use. In this case, I've used BLAST. But yesterday, you saw an example by Kim Pruitt from the NCBI. And she said that you can also use their tool, which uses Kamer seeds. I think it was called FCS. So you can use many tools. There's Tiara, there's Minhash, there's Kraken. There's many tools that you can use to do the taxonomic assignment. By default, we use BLAST because that's what everybody uses. So we color the contigs based on their BLAST matches. And that's what the colors look like. So the blue contigs, the light blue contigs are Ascomycota, which is a phylum of fungus. So that's good. That means this is our target organism. Great. The dark blue contigs, which you can't see so well because they are, the color is not so obvious, are no hit. There are 4 of them. I don't know where the 4th is. I think it's over here. And there are 3 contigs, which are proteobacteria, which total 4.5 megabytes, and they are separate. So what this blob plot is showing in one glance is that, yes, you have sequenced your fungus, that's over here, but you've also accidentally sequenced a proteobacteria, quite a lot of it, 4.5 megabases out of 41 megabases total. So a blob plot shows you all of these things visually at one glance. And obviously, I've picked a very clean example. I will also show you some bad examples after this. So now I'm going to go into the website and the demo and the viewer. The main thing to remember with Blob Toolkit is it has 3 parts. There is a viewer. That's the interactive website that I'm going to show you today. I'm not actually going to show you the other 2 parts, but I'll tell you what they are. So the viewer is something that we have created for all public INSDC genomes. By INSDC, we mean genomes that have been submitted to NCBI, and that means it is also available in the European Nucleotide Archive and in the Japanese databases. So it's available across the world freely with unique accession numbers like Kim Pruitt told us yesterday. So the viewer is designed to give you an insight into all the public genome assemblies. The one I showed you just now, this one is also a public genome assembly. Remember what she said, that they try to filter out the contaminants, but they don't always succeed. And what our tool does is it lets you see the public assemblies and it lets you see where the contaminants are, so that if you use those genome assemblies, you don't accidentally make a claim which might be wrong, because you might say, oh, look, this genome assembly has these amazing genes which do this. And later on you realize, oops, those are actually coming from the bacteria. The reason why they separate out as blobs separately is because different organisms are expected to have different coverage. Notice that all the contigs of the same organism are clustered closely because they all have roughly the same GC and roughly the same coverage. And over here, it's a very different coverage. This is about 10, this is about 16. The GC is very different. This is about 68%, this is about 53%. When you see such a big difference, you know they are separate organisms. I'll show you examples where you don't always see such a big difference. Anyway, so that's why we make blob plots. And the viewer is what I'm going to show you today. The other 2 things that are on the Blob Toolkit website is the Blob Tools 2 download and command line tool for creating the data format for your own assemblies. Obviously, you don't only want to analyze public assemblies, you also want to analyze your own assemblies. And lastly, which goes along with Blob Tools 2, is the pipeline. It's a snake-make pipeline for running your own analysis. It's not easy, but you have to be familiar with the Unix command line and you have to have access to a cluster with lots of RAM and lots of cores, because this is a computationally intensive process. That's why we run it for all the public INSDC genomes, but we also show you how you can do it for your own genomes when you are creating genomes for the Africa Biogenome project or for the Daventry of Life project or for any other project. So now let's get on to the demo. How much time have I used up? Right, lots. I'm going to go straight to the demos. So I'm going to click on this and it takes you to the Blob Toolkit website. Like yesterday, Kim was saying, a live demo is always a bit scary, but we'll try it. So we go to the open viewer. And the first thing you see is an option to search for different data sets. I am going to come here and say, let's look at the Didymella arachidicola data set. You can also click on this, but I'm going to show you Didymella arachi. Oh, it completes. Great. Click on that. It brings you the same plot that I showed you just now. A couple of things. You're not seeing a very important part called filters. I'll click that, filters. And now you can see a whole lot of different options. There's the blob option, there's BUSCO, which shows you all the BUSCO scores for each different organism. So if you do this, it shows you the BUSCO score for the overall assembly against different databases. So we pick the most specific, then we go up Ascomycota, which is the phylum, then we go up to fungi, then we go up to eukaryota. We also check bacteria because that is a way of checking if there are any bacterial genes in here. And we can see there are lots of bacterial genes. We already know that there's proteobacteria. And lastly, we see archaea. So we do BUSCO against everything. So rather than just doing BUSCO against one data set, we do BUSCOs against all relevant data sets. I'm not going into details of these other views on top right now, but I'm going to show you the table view. The table view shows you all the contigs, their length, their GC, their coverage, and it shows you what assignment we gave it. We gave it Ascomycota or Proteobacteria. And what you can do, sorry, I'm just checking my timer again. There we go. And what you can do is you can now say, how was this assignment done? Why did we call it Proteobacteria? So if I click on that Proteobacteria, it shows you for that contig, which is quite long, it's 3 megabases long. There were many, many, many hits across the whole contig. And those hits are where all the BUSCO genes were, because we don't want to take the whole contig. We only want to take the informative regions. So we take them and we blast them, and we see that most of the hits are to Proteobacteria. So that's how we know that this entire contig is Proteobacteria, because most of the hits are to Proteobacteria. Another thing you can do is you can actually see which Proteobacteria. So I go down here on the left, and we see it says BUSCO genes phylum. But if I, apologies for the noise, I'm in India and it's quite noisy, as you might expect. If you click on BUSCO genes, where are we? BUSCO genes, genus, BUSCO genes genus. Now you see there are lots of things, and it even shows you the genus, which is Strenotrophomonas. So most likely this bacteria is from this. If I click on this category, don't worry about, following along. You can follow YouTube tutorials and stuff that we have made on how to do this. I'm just giving you a demo. If I now click on this, it shows you that most of the hits in that contig are to Strenotrophomonas. So we are quite sure that this is a Strenotrophomonas genus bacteria. And similarly, all the others are Ascomycota genus, which is the closest genus, because it's not the main genus that we did. Anyway, so that was the main point I wanted to make about this. I'm going to go back to my slideshow. The next example is Casuarina equisetifolia. It's a tree, and it's quite a good assembly. Let's click on it and see what happens. Now, the first thing you might notice is that it does not have those nice circles. The reason why is because for some of these assemblies, we realized there were many, many, many thousands of points. It's a bit hard to see. I'm going to zoom in. It says total 2,900 points. So rather than seeing 2,900 circles or contigs, we decided that we would show it as a binned histogram. That means there's bins of square shape. But if you want, you can change it to the circle view, which is easier to read, I think, in my opinion. So we go to settings, we go down to where it says shape, and instead of square, which is the default right now, we pick circles. And now you see, we're seeing something very similar to what we had last time. You again see a whole lot of green circles, and you see this sort of one blob of green over here, a second blob up here, and another blob of blue over here. And here's a funny thing. You also see a whole lot of blue down here, which is zero coverage. It's a log scale, so we've just put it on the log scale, but essentially it's zero coverage. So now you might think, what's going on over here? This looks like our organism of interest, because you can see the green color says Streptophyta. Streptophyta is the phylum which has this tree. And this blue over here is Ascomycota. It's a fungus. So this tree sample, again, taken from INSTC, taken from NCBI, has the tree in green, and it has all of this fungus, and it has something else which is down here. So here's a cool trick that you can do in Blob Toolkit. You don't always have to have only the GC axis on GC on the x-axis. You can put other things on the x-axis, such as, let's go to filters. And if we scroll down, you can see there is coverage. This coverage is from a read set that is available on NCBI. So SRR means it's a SRA read set accession number. And there's a second read set. So for the same species, they sequenced 2 libraries. You can have multiple libraries for the same species for the same assembly. So we take that second species, and this one right now says there is no axis live. None of the axes are highlighted. If I click on this, it will plot the coverage on the x-axis of this library. I'm going to do that. I press x-axis. And suddenly you see what you would expect to see is all of them should have been along a diagonal. But what you are instead seeing, because if both libraries have the same organism, you should see it at the same GC and same coverage in both same coverage in both axes. But because in this library you have 0 coverage, and in this library these have 0 coverage, that means these contigs are not part of the main organism because they are completely absent in one of the libraries or the other. And now what we can do is we can actually select these contigs and make a list of them, so we can say, please remove these. So how do I select? I can just click over here, define this region. I'm going to try that. You can't click outside the plot because that resets the selection. I click over here and I click here. So when I finish selecting, it highlights those contigs which do not belong to the target organism. And it says, we want to remove these. We also want to remove the other blue things, but these ones we definitely want to remove. So how do we remove it? I now go to the selection filter, which is right now it's off. So when I put my mouse on it, it says activate, which means it will turn purple. And when I activate it, it only selects those contigs and it makes the others disappear. So now that they've all disappeared, this is great because now we can go to lists. And it says, overall, you had 2,936 scaffolds or contigs. And right now you have selected 2,200 scaffolds. So many scaffolds have to be removed from this assembly because they have 0 coverage in one library or the other. And so it's very easy. I'm going to give this a name. I'm going to call it 0 coverage. I'm going to say create a list. So it's created a list called 0 coverage. So these scaffolds have to be thrown out. And if I click on this, it shows me the list. I say, give me the full list. It downloads it. I click on it. It's a JSON file, so it opens up in Firefox on my browser. And you can see it's given me the names of all the contigs that have to be removed. So if I now take these 2,273 contigs and I remove them from my assembly, I will be cleaning up my assembly. And that's how I know that these do not belong to my target organism, which is the tree. I hope that example was clear. I'm running out of time. I'm not going to do the last example. So my last example was the tardigrade with many contaminants. I'm not going to go into detail, but I'm just going to quickly show it to you because it had so many contaminants. This is the one that I just showed you the paper. I'm going back. This paper, the one that I'm going to show you just now, is a public assembly. It is submitted to NCBI. And that's the assembly that was used in this first paper. And I'm now going to show it to you. Just quickly, just so that you can see how many contaminants it had. Okay. By default, it shows it with that 2-dimensional histogram. I'm going to go to settings. I'm going to go to shape and click circles. And right away, you see, this is the main blob where the tardigrade is. This was done using short reads, so it's a very poor assembly with lots of little circles. But look at how many different colors and blobs there are. This is a separate bacteroidetes. This is a separate bacteroidetes. This purple is something else altogether. It's showing up as other over here. This green stuff over here is proteobacteria. The orange stuff over here is chloroflexi. I mean, this thing was full of contaminants. And you can see that there are different coverages. There are different GC, they're totally separate, they are not part of the genome. Once we removed them, we were able to show that the actual genome assembly of the tardigrade does not show extensive horizontal gene transfer. So I'm going to stop over here. I'm not going to explore the plot more. These are my slides in case the live demo didn't work, so I'm going to go through it. So in summary, what have we learned today? You can go to data sets. You can search by taxonomy, by taxon name. You can even type in the assembly ID if you know it. You get many fields. You can sort by them. I've shown you 3 examples. I've shown you some views. You can do blob. You can do table. You can do buscope. In settings, I showed you how you can go to the blob shape. I didn't show you axis and static, but you should click around and try it yourself. And in filters, you can drag the numerical filters. You can change different coverages. You can change the taxonomies. I didn't show you full examples. This was from a slideshow I had done for a 1-hour-long workshop. I'm going to be quick over here. And the selections allows you to change which parts of the blobs you want to select. And then you can make a list for removing it or for keeping it. And that's how it works. So last slide. next steps. So you have access to this presentation. So if you click on this, you will see all the online resources. The website is being fixed. You can report issues at our GitHub. There is a YouTube channel with small videos, including an earlier video with a longer demonstration. And the remaining slides in this presentation are actually quite useful if you want to do them on your own. So please try them. The presentation is publicly available at the URL that I gave you at the start, and Taiwo will make it available. So lastly, we've made it at the University of Edinburgh. Richard Chalice is the main developer. He's the brains. Mark Blackster is the head of the project. He wrote the funding applications. I helped develop it. I came up with the idea, but I didn't do the programming. I'm not such a smart programmer. And people at the European Nucleotide Archive and the European Bioinformatics Institute were amazing. They helped us integrate this into the ENA website. There are links over here. All our funding agencies are over here. And thank you very much.
Yeah, thank you so much, Sujai, for the presentation. Very interesting. So, but you will be on the call, but we'll proceed to the next presentation to take now. And so would like to ask Tiange to unmute herself and proceed to make her presentation. Is Tiange on the call?
Who is it? Taiwo?
Yeah, Nigi.
Nid.
Yeah.
I haven't seen Nidhi. Nidhi,
Okay.
if you're here, could you please? So Nidhi, I don't think. Tiagi,
Okay. Yeah.
I don't think she's here. Just a moment. I'm just gonna chat her up. She did chat me up.
Okay.
So actually, she's not online.
Okay.
Yeah. So maybe
So,
you want to get some questions from Sujai.
yeah,
Looks like he has a lot to answer.
Yes, we can use this bit of time to ask questions. So let's ask our questions and directly to Sujai if we can receive answers to them. Does anyone have questions? You can raise your hand and then you can ask a question. But while people are raising their hands, I would like to read out questions that are posted on the chat box. One question is, please, my question is, to do real bioinformatics, is it recommended to work under Linux or even Windows? So that's one question. Yeah, if you can go ahead and answer that. Okay, so the second question is, if you are using, okay, still more or less in the same type of questions, but now is on the blob toolkit, and the question is the blob toolkit applicable to only limited species? That's a question from the oh. Hello, Sujai, can you hear me now? I've unmuted you.
It takes a long time for the message to come through. You might have said unmute, but I don't see the message until 5 seconds later. That's why.
Oh, my apologies.
Anyway,
okay,
no
I'll go to ask the questions again.
I'll do the first question.
Yeah, the first question is, to do real bioinformatics, is it recommended to work under Linux or even Windows?
So I think everybody on this call who has done bioinformatics will tell you that you should do it on Linux, because it's the one that the maximum number of tools are written for, and it gives you the most flexibility and power to do new tools and things. You can use, I actually use a Mac laptop, but I use it to connect to a Linux server. So it doesn't matter which machine you use for connecting or for small tests. You can do it on a Mac, you can do it on a Windows, you can do it on Linux, but I think heavy bioinformatics is best done on Linux computers. But that's my opinion. I think other people will tell you other things. But the most tools are written for Linux. Nowadays, more and more tools are being presented through Conda and Mamba. And so you can get them on Mac and Windows, but sometimes they don't work. So definitely Linux should be your first choice. There is a learning curve, but I highly recommend it. Does that answer the question? Just write it in chat.
Yes. And the follow-up question to that is, if you are using a Linux interface, does it still require large memories to accommodate the complex data?
So in Blob Toolkit, yes, because we are doing large blasts against the entire NT database. But you can do other things. You can not use blast. You can do other things. You can use the FCS tool, which I checked yesterday, the tool that Kim mentioned. And it also requires a lot of memory. She said about 500 gigs of RAM. So yes, you do need access to large servers. I realize that it's hard to get machines with large RAM because it's expensive. But companies like Google and Amazon, and I think even some nonprofit organizations in the US like Cyberse, CYBERSE, will give you access to large machines for short periods of time for free. So definitely explore other options for getting access to clusters. Even if you don't have the big machine, you can connect to the big machine and do all your data there. So you do need a lot of RAM for some applications. For other applications, like nowadays, Genome Assembly can be done quite easily on less RAM. But things like Blob Toolkit and other tools like FCS that Kim mentioned yesterday do need a lot of RAM.
Thank you. More questions coming. One of our participants is interested to know if the Blob Toolkit is applicable to only limited species.
It's applicable to all species because the GC and coverage, it is completely species agnostic. It does not matter what species you put in. If it's 2 different organisms, you will usually see them, or 3 or 4 or 5, you will usually see them at different coverage and different GC. The coloring of the contigs by taxonomic assignment depends on what other contigs are already present in the public databases. That's why we don't give you just the genome assignment, the taxonomic assignment at the level of the species or the genus. We take it all the way up to phylum, so that you can even, or even superkingdom. Like maybe all you can tell is that it's bacterial and not eukaryote, nothing else. The point is you can use any tool. We use BLAST by default, but the Blob Tools 2 command line lets you use any tool which can do taxonomic assignment, for example, Kraken or some other thing. So there are many tools. I can give you details. Somebody can contact me. But by default, we use BLAST because that's what everybody understands. But actually, you can use other tools which are not limited to any species.
Thank you very much. So we have Negi Tiangi on the call now. And we are going to proceed to take the next presentation. And thereafter, we can ask the other questions, all the questions together, and we can get answers to them. So, Negi, you're welcome to the call. Please proceed to make your presentation. I have unmuted you.
Hello, everyone. Am I audible?
Yes, you are.
Hi, everyone. My name is Niji Tyagi. I work at Uniprot. I am giving this presentation on Uniprot with my colleague, Kate. We're sitting at, so let me share my screen. So I work as a scientific database curator at Uniprot, and we are going to talk about our database Uniprot, which is Universal Protein Database. I am presenting the slides about our database, and my colleague Kate has helped me to put all this slide together. So acknowledgement to her. So Uniprot database is based in EMBL-EBI. The European Bioinformatic Institute is a part of European Molecular Biology Laboratory, which focuses on research and services bioinformatics. It is located in Welcome Genome campus in Cambridge, UK. EBI provides more than 40 open data resources, which are used regularly by millions of scientists around the world. EBI has databases services for chemical molecules, drug discovery, genes, genomes, RNA for proteins, for genetic variation, diseases, and literature. We at Uniprot fall into the category for protein databases services alongside PDB, PFAM, ProCite, and Interpro. Uniprot is a consortium. It has 3 members. Let me remove this thing. Yeah. That's it. Yeah. Apologies. So Uniprot is a consortium. It has 3 member states, 3 members, which contributes towards it. It is a collaboration between EBI in Cambridge, UK, Protein Information Resource PIR in United States, and Swiss Institute of Bioinformatics, SIB in Geneva, Switzerland. Uniprot primarily is a resource for protein sequences and their function information. We provide comprehensive, high quality and freely accessible data. We have protein sequences, including their isoform, disease variants, and post-translation modification. Every cross-reference available for a protein sequence is also provided. So user can come to Uniprot and can navigate all the information available for that protein in other databases. Uniprot comprises of 4 sections optimized for different users. First is Uniprot knowledge base. Second one is species, proteomes. Then we have protein clusters, and then we have sequence archives. So this slide talks about the first section, which is Uniprot knowledge base. The Uniprot knowledge base consists of 2 sections, Swissprot and Tremble. Swissprot consists of manually annotated high quality records. Curator extract information from scientific literature. Curator also carry out computational analysis and evaluate the result to extract information for proteins. We have more than 500,000 of reviewed protein entries. Tremble, on the other hand, consists of unreviewed entries. These are proteins for which the annotations are added computationally. Currently, we have nearly 226 million sequences in Tremble. The second section of Uniprot is proteome. Proteome, as we know, is a set of proteins which are thought to be expressed by an organism. Majority of these proteomes are based on genome sequences, which are submitted to ENA, GenBank, and DNA database of Japan. To supplement the proteomes, we have developed pipelines to import genomes, which are sequenced and annotated by ensemble, vector-based, and worm-based parasite. We also import proteomes from RefSeq, especially which are annotated by NCBI eukaryotic genome annotation pipelines. We have more than 20,000 reference proteomes, and depending on the importance of the organism, we have higher proportion of these proteomes manually curated. For example, all the proteins from Baker yeast and Escherichia coli strain K12 are manually curated. The third component of Uniprot is Uniref. Uniref provides clusters of protein sequences, which are grouped based on their sequence identity. For example, Uniref 90 consists of protein which are grouped based on 90% sequence identity. Clusters are important, as we know. If we know the function of one protein in a cluster, all the other protein can be annotated based on that function for that protein in that cluster. The last component of Uniprot, The fourth component of the Uniprot is UniPark. It contains all protein sequences from public protein sequence databases, including historical obsolete sequences. This slide actually shows the growth of data we observed over time in Uniprot. We see there's exponential growth in our database. We can see suddenly there was a dip in number of protein around 2015. During this time, we had applied redundancy removal process. This process identifies and removes almost identical proteomes of the same species. Removing redundant sequences, it greatly reduced the size of Uniprot KB. It also made its growth more scalable. And we can see here, the bottom of the graph, we have around our 500,000 manually created entries, which are part of Uniprot KB. So this slide talks about how we do manual curation at Uniprot. Uniprot creator read the papers and extract relevant information from the literature. This information could be related to various features of proteins, such as function, catalytic activity, pathway, interaction, tissue specificity, any biophysical, chemical properties, and so on. Curator annotates proteins based on information in the paper. If a direct evidence of, say, activity of protein is given, curator mentioned that on Uniprot page. In case of direct evidence, a curator can also derive information based on existing evidence for similar proteins. So just to show an example, this is P53 human protein. If we go to this page on Uniprot, if any data or information is given from various papers or any paper, then tags for all the publications are given. So any information on this page, it will tell you where or which paper this information is coming. Sometimes, as I said, there is no direct evidence. Curator can still assign some annotation based on similarity and experimental data for related proteins. How a curator made this decision is also tagged for such annotation. For example, here for this protein, some function is annotated to this protein based on similarity. And this has been put by a curator based on the similarity for this protein sequence. So this is also tagged, so you can know from where this information or from where this tag is coming. And of course, if you think there's a gap in the information, you can, of course, let us know and we can review if we have to add or delete some information from the page. We also link our protein to various external databases, such as enzyme and pathway databases, protein family databases, 3D structure databases, phylogenetic databases, sequence databases. You can just click on this external link and you can find this protein in other databases. You just have to, and all the comprehensive information, the link to other databases is present on this external link. So we talked about how manual curation is done in Uniprot. There's also other part of annotation, which is automatic. And it is done primarily by two processes in Uniprot. One is called UniRule, and the other one is called ARBA. So in case of UniRule, expert curators, they set condition for these rules, and after carefully selecting the rules, applies annotation to the proteins. So there's manual intervention in these kind of rules. These rules are supervised. We have expert and dedicated curators who constantly derive new rules, amend them, tailor them, and apply them to assign annotation. On the other hand, we have ARBA, which are completely automated rule system, which is based on machine learning. These rules are also trained on Sysprot entries. It predominantly utilizes protein domain classification tool Interpro. These rules are not supervised by curators. Both can predict protein name, catalytic activity, function, subcellular location, or pathway membership. UniRule, the first one, can annotate sequence-specific information such as PTMs and active sites. So this is an example of UniRule, automatic annotation. These rules are looked after by curators. For example, if a sequence has signature as a kinase domain, ATP binding region, active site signature, map kinase signature, kinase domain profile, or catalytic domain, and it comes from eukaryotes, then these annotation can be applied to the other unannotated proteins. By using this rule, we could annotate nearly 12,000 proteins in Uniprot. And this is just a list of, if you click on that 11,763 entries, you get list of all the proteins which are annotated by this UniRule. And this is a simple example of ARBA. This is if a protein has these interprodomain for hemolysin, and if the protein is from bacteria, then a function of hemolysin is associated a protein from bacteria. We could annotate nearly 150 protein in Uniprot KB from bacteria using this rule. Then when come to something called unifier, you can annotate your own protein by using unifier. You can feed your protein sequences to unifier engine, and it will predict annotation for your protein. Unifier tool and its documentation can be found on our GitHub page. We also have a webinar on our training pages which explain about unifier. On our website, there's also provision for community curation. If you think some paper of yours or some other literature should be added to a protein, you can do that. You can go to any protein page, click on add a publication, and you will have to sign with your ORCID ID. This is just to validate the submission is genuine. And once you have added some information from a paper, you get credit to add that information on Uniprot page. You get credit for that curation. There has been almost 3,000 community submission till now, relating to 1,100 papers from nearly 500 unique users. If we go to just Uniprot homepage, uniprot.org, there are different tools which are available on Uniprot homepage for searching your sequence using BLAST, doing alignment, mapping different protein IDs, or searching for small peptides. Our homepage also gives access to FTP download of all the data. It gives programmatic access. Researcher can also submit data, which will be reviewed by curators and will be included in the database. There is something called advanced search in the database, which is very useful. From the many drop-down boxes, user can select any feature, and search function will list all the proteins satisfying all the conditions set by users. For example, if a user wants to look at kinase protein, which come from Homo sapiens, human, which also have a 3D structure and which is also reviewed. So they can search using this drop-down search engine pattern. If you search this, then it will list all the proteins which are kinases, which are from human, which have structure, and which are also reviewed. So this is a very simple example I gave here. It's a very powerful tool. I would really request everybody to just use it once, and it's pretty awesome. We also have BLAST, like a search engine on our Uniprot homepage where you can go and search your sequence. The important thing which I want to tell here was that when you get BLAST result, you can also select to see any important residue you wish to see in your query. For example, once you get the alignment to one subject or a hit, you can choose some specific annotation or feature, and the BLAST result, it will show those residue which are of interest to you. For example, if you choose active site residue here, it will highlight those residue in the alignment. And then from there, straight away, you know whether that residue is conserved in your protein or not. Uniprot also provides several APIs to query and access its data. All this information is present on our homepage. If you have any query about it, please write to us and we'll get you in touch with our developers who will be really happy to help with any query regarding API. We have also extensive help pages for any query you might have for Uniprot. We also have many training material present on our Uniprot help pages. And this is our help desk email ID. If you have any query regarding curation or automatic annotation or regarding APIs, you can write to us. We also have a YouTube channel for Uniprot. There are tutorials for exploring various features in Uniprot. Various curators and developers have prepared these slides and presentation so that a user can get an idea what we do and how we do it in Uniprot. On our homepage, you can find training videos and various online courses directly. And this is the list of staff members who work for Uniprot team across 3 sites. And this is the list of staff members. We thank our funding bodies. There are many, many funding bodies and organizational institutes who support Uniprot. So I thank you for your time and attention. If you have any question, we'll be happy to answer them.
Yeah, thank you so much, Nigi, for the presentation. But we hope you'll still be on the call, but we'll take the next presentation thereafter. Questions that are directed to you can ask you.
Sure.
Thank you once again. Okay, now we are going to the next presentation. This is going to be taken by Tim, and I believe that Tim is on the call. Would like to give you the floor now to make your presentation. Nigi, please, if you just stop sharing your screen. Just, is Tim on the call? Yeah, I can see Tim on the call. Yeah, please, I've asked you to unmute.
Yes, okay, I'm finally unmuted. All right, let me go to screen sharing. Okay, you should be able to see my first slide.
Yes.
Okay, thank you, everybody. So our presentation is going to be different from the ones you've seen just now. This is not a technical presentation. It's an opportunity for us to introduce ourselves, tell you who we are and what we do. And just so you also know, we've only very recently become affiliated with AfricaBP. So this is really a good chance to get to know what the community looks like and what your interests are. And hopefully you'll like what you see from our presentation today, and we'll continue with the dialogue. So this presentation will be split into 3 sections. I'll be starting off with an overall introduction to Diplomics, and then Shane Murray will talk specifically about some of our initiatives in the genomic space, and Padamoyo will do the same with Bioinformatics. So Diplomics is short for Distributed Platform in Omics. We are a research infrastructure, which is a name given to an EU-style government-supported setup in specific technology areas. And we happen to be one of 8 that have been launched by the South African Department of Science and Innovation. And we are the only one specifically within biotech. And even in that space, we're focused specifically on genomics, proteomics, metabolomics under that, and bioinformatics. So I usually direct people to our website. This is where you can get a comprehensive overview of who we are and what we do. And there are a lot of links within our website that can take you to other platforms. We have extensive collection of videos that we produce. And we also have a presence in social media on LinkedIn and Twitter and YouTube. All of these are very easy to find if you just search on the word Diplomics. So we are a network. So our approach to a research infrastructure in the omics space in South Africa is a network model where we have we are currently working with a number of pre-existing labs in South Africa. The partnerships or the network labs are part of Diplomics under several criteria, one of which is that they might be a research lab, but there's some component of services. So one of the driving aims of Diplomics is to create a broad service community for students and researchers who want to pursue omics technologies. So we have currently about 8 genomics labs, 5 proteomics labs, and 5 bioinformatics groups. But Shane and Potamoyo will expand on that in a bit. But we are a growing network as well. And over the last year, we've added new affiliates to our program. And I'll explain in a moment what that means and what's behind that. So the best way really to explain our program is to tell you about what our objectives are and the actions we pursue to fulfill those objectives, and also the impacts that we are pursuing to, as a government-funded entity, to demonstrate our value to the broader community. So we have 2 objectives really at a high level. One is to build a world-class omics infrastructure in South Africa. And we have a number of specific actions to do that. So that includes buying and placing instruments in our network system. We also support service and maintenance contracts on those instruments. We found in the beginning of our program that many, very expensive and important instruments had no service or maintenance contracts. And so many of them were dormant for simple reasons because there was no budget to either repair it or maintain it. And along with that, we sometimes also look to support extended warranties. And then very closely affiliated with that is the advanced training of technicians. So obviously instruments are no good if you don't have the skilled and experienced people to operate and to run them. And by this, we also mean not just operating the machine, but also to be able to conduct the up and downstream aspects of omics, such as sample preparation, project design, quality control, preparation of few samples, but also the analysis of those samples afterwards. So impacts are quite important for us because it's part of how we are measured against our funder, but it's also important to show that we are aligned with a lot of the efforts of other groups. And I've already seen in some of the presentations yesterday that we're very much aligned with the same kind of goal-oriented impacts in African omics. So one of the things we do further in identifying impacts is actually break them down into different types of impacts. So we look at, at what we call direct and induced. So these are impacts that can be measured or identified early on. So for example, if we place an instrument in a lab, that lab now has an ability to perform some kind of research or service. If that machine is used to help generate revenue in a sales model, then that's a type of induced impact. And again, those are fairly immediate or short-term returns on investment. The other parallel track we pursue is to enable access to students and researchers to omics infrastructure, skills, and technologies in the country. And we do this through a lot of training. So we initially started off, as I said, in the upper grouping of actions, advanced training for technicians, but we've now greatly expanded that, and I'll be touching on that in a moment, really to span everything from high school outreach up to very advanced bioinformatics training. And then we have a program called Spatial Transformation, which I'll talk about in a moment, but it helps to address the conundrum we faced in the fact that in a South African environment, and this is probably true in many places in the world, where building a world-class omics infrastructure really requires having the enriched environment for that to take place. So you need the building to put your instruments in, you need the technicians, you need reliable electricity and water and all of these other elements, plus all of the ancillary equipment that you need. And those are really best located in top-tier universities or science councils, and in some cases, private companies. Yet a big mission of ours is to, like I said, enable access to students and researchers. So spatial transformation for us is a form of transformation where we're taking omics out to all of the regions in South Africa. And like I said, I'll touch on that in a moment. And then a third component of enabling is that we are recently supporting high visibility and high impact projects. And I think this is where we see a lot of exciting potential in working with the AfricaBP Genome community. And these are projects where in many cases, we see that many of the elements needed to take on projects are in place. You've got the instruments, the training, and all the other infrastructure, but there's no money to actually pursue the project. So in very select and specific cases, we are supporting projects that are highly visible. So it's something that we can promote and make people aware of, but also has high impact. So obvious examples would be things like, precision medicine for high burden diseases, or it could be towards biodiversity conservation. And then the impacts we measure here are more indirect and in many cases even cumulative and can take months or even years to be felt. But the outcomes here are developing skills, increasing awareness about science and technology broadly, but omics specifically. Again, improved access and increased opportunity for many more people to be able to get into omics research. And then finally, world-class research is conducted here. And very importantly for our funder, the Department of Science and Innovation, is to see reputational gain. In other words, to say, we can do the same world-class omics research here as anywhere else in the world. And then threading through all of these is media. Now, media here really can mean everything from, again, visibility and awareness. It could be publicity, but it's really a very strong component of what we do to get people to understand a very esoteric field, really, which is generally not that easy to explain to people. So we've put a lot of time and effort into developing media that we can use to help propel our message. So for example, on our website, we have several series of videos that we've developed ourselves. The first round were profiles of all of our partner labs. So I encourage you to go see those. These are relatively short 2 to 3 minute videos, but helps to illustrate what that particular lab is doing and why it's of value. So the very first one in the upper left-hand corner is a profile of one of our smaller labs that does genomic services in the agricultural industry. And so what we showed is how much of an impact this has in food security, for example. We also have a YouTube site where we have many more videos, and the most recent series of ours are Women in Science, which took place in the month of August, and then other sometimes technology-specific videos or demonstrations. So I encourage you to check those out. So one of the things about our program is that we strive to be innovative and to be highly efficient. And one of the things that we've learned in Diplomics over the last 5 years is that a lot of the omics activity in South Africa, at least, is quite inefficient. And one of the main reasons for this is that almost an ironic situation where it's relatively easy to get funding for instruments, but very difficult to get money to operate those instruments. And operate can mean the repair and maintenance, but it could also mean being able to conduct research on those instruments. And from a budget perspective, we're also lean and efficient. We spend nearly 85% of our budget on direct initiatives to support omics in South Africa, and a relatively small proportion goes to management and to things like brand management and business development. So on the right are just 2 examples of some of the approaches we're taking. One of the things we also do is technology showcasing. And most recently, we've been focused a lot on the Oxford nanopore systems, and in particular, these Mark 1C devices. This is an example of one from one of our labs recently. And the idea here is to really expand access to being able to do omics. And these devices are brilliant. They're 1/1000th the cost of a NovaSeq platform, and they're completely mobile and relatively easy to use. Not high throughput, but you can do whole genomes on these things. And Shane will be expanding quite a bit on how we are using these in our programs, in particular within the Spatial Transformation program. And then on the proteomics side, we also have been the first group to bring in a technology called Olink. I know that's not too relevant to this group today, but it's an example of where we can use our funding and our mandate to bring in new technologies. In this case, Olink is a way of identifying proteins that uses next generation sequencing technology. In this case, particularly using an Illumina platform, but it's the first instrument in Africa, and it's first cross-omics type of instrument. A very large component of what we do is human capital development. In fact, this is mandated by our funder to be up to 30% of our budget spend. So we very recently conducted first of what we hope will be many high school outreach programs. We work with NGOs, in this case in Comba Youth, where we brought in high school learners who were shown a demonstration of how to extract DNA from strawberries. And there was a bit of a light level lecture beforehand on what is DNA, et cetera. But really the aim here is just to give these kids an exciting and interesting way to see the value of science and technology, and really to hopefully inspire them to become scientists themselves. And probably like many countries around the world, there's always a shortage in the pipeline of young people coming into science and technology. We have a standard contract with Thermo Fisher Scientific to provide more higher level, more complicated and more technology specific training. And we do this throughout the country. It's a very good situation where we can use their deep skills and experience, and also their broader footprint in the country to be able to deliver training on our behalf. On the right side, we've also supported recently what we call a Teach the Trainer program. So in South Africa, many biology high school teachers have not really been exposed to the kinds of science we'd like to see in omics. So this is also an example of, you know, half a day lecture, half a day hands-on DNA extraction, but it has a wonderful multiplier effect, and that these teachers with their enthusiasm go back to their classes and help infuse you know, passion in their students for science. And then finally, we've been sponsoring and working through new nodes added to our program to do specific training. In this case, on the bottom right, it's with the South African Aquatic Institute for Biodiversity, and this was a course on 16S metagenomics. So I mentioned spatial transformation earlier. In essence, what we want to do is take our essentially, you know, 3-location structure. This is Western Cape and Gauteng and Durban, where most of the higher end universities and the infrastructure exists, and find ways to spread that and get it out to the rest of the country. I think what's quite relevant and hopefully of interest to you is that some of our sister research infrastructure groups are involved in an array of environmental observation networks. And this, so these are, sorry, I just can't see my own slides here, just a moment. Yes, so these are under the National Research Foundation support groups. So we've got shallow marine observation groups, a recently launched research infrastructure in polar studies, and then also the South African National Biodiversity Institute. And there were some attendees yesterday. I think they're here today as well, also from SANBI. So again, we're still a relatively young program. So developing these partnerships is exciting. And should give us a lot of opportunity to branch out. And so just quickly as an example, the shallow marine and coastal research infrastructure has mature and established site locations along the entire coastline of South Africa. And they do a lot of sampling of the marine organisms, which they really are keen to add an omics component to that. And also a newer program, the FTON, which is the Environmental Observation Network, has set up these nodes across different particular ecosystems in South Africa, which we can also partner with to set up projects which would be using, for example, nanopore technology to do environmental sampling, for example. So I just want to shift gears a little bit. I'm almost done here. I mentioned this yesterday in my question to Andre from Illumina. And one of the things I think is very important to achieving the goals of Africa Biogenome, and I recently read your comment piece from Nature, is the ambition to sequence 105,000 genomes and what that is going to cost. And it's a staggering number. And one of the things for us that we have identified as a significant challenge, and this is not something we've just come up with, but it's what people tell us all the time, it's just the prohibitive cost of doing, in particular, short read next generation sequencing in South Africa. So on the slide here is a press release from August of 2020 when Illumina announced that they have now unlocked the $600 genome. And I really thought, you know, this was a disingenuous statement to make, because that's just the price of the kit, and that's the price in the United States or in Europe. It doesn't factor in the cost of all of the other things you need to be able to do sequencing, especially the cost of the instruments, but all the other things I talked about, the ancillary equipment and the technicians and the building, et cetera. So recently, one of our partners has worked out what it actually costs to do sequencing currently in South Africa. And this cost is more than double the stated cost that Illumina has proposed. It's currently about $1,300 in South Africa. That's about 22,000 rands. Yet this still doesn't include the cost of the instrument. And we've done that purposefully because most labs in South Africa get an instrument through a grant or a donation, and they don't have to factor that into the cost. But nevertheless, even without that, you can see that we're starting off with a price penalty of nearly 100%, actually more than 100%. So one of the ways to get around this problem, or to get it, I guess to surmount this challenge, is to go around it. And this is already not new technology, but the Oxford Nanopore systems, the Nanopore Mark 1C I just showed you, seems to us to be really, without exaggerating, a game changer. It's going to allow us to buy many, many machines for the price of a single Illumina instrument. I'm not bashing on Illumina, by the way, but it's just a reality we have to face. That is very difficult for people to get into omics looking at those costs. With these devices, on the other hand, we can buy dozens and dozens of them, train people on how to use them, and get them out to places where they're needed. And I think this is an excellent way to get ourselves involved with the Africa Biogenome project in sequencing genomes. So that's it for my part. I'm going to hand over to Shane Murray, who will take you through a few more slides. And Shane, just let me know when to advance the slide for you. You might need to unmute Shane.
Yeah, I will do that in a moment. Yeah.
Shane, can you hear us?
Yes, hi. Thanks, Tim.
Hi.
Thanks, Hawa. So hello everyone from Cape Town. My name is Shane Murray, and I'm one of the genomics coordinators in the Diplomics network. And I'm just going to take over from Tim now just to talk a little bit more about what we are doing specifically in the genomics network, which forms part of the overall genomics program. So Tim has already shown you this picture, which shows all our partner labs throughout South Africa. And this is just to illustrate from a genomics point of view, we represent a diverse number of different types of NGS technology. So of course, Illumina leads the pack. We have 6 different labs with Illumina technology, and this ranges from labs with MySeq all the way to labs with NovaSeq. We have 1 lab in the network that has an iron torrent, that is specialist in iron torrent sequencing. And then we have 2 labs that have MGI sequencing. And what we have established over the last year is what we are calling our Nanopore working group. And I'll be saying a little bit more about that on the next slide. And as Tim has already stated, we really have 2 overall aims. One is to connect researchers and postgraduate students in South Africa to world-class NGS facilities. So to that end, we are promoting the infrastructure in all of these different genomics labs so that people can send their samples there for research purposes. And our second overall aim is then to enable training and access to next generation sequencing, particularly to researchers and students who are in rural institutions or in previously disadvantaged research institutions. So as Tim has pointed out, there are hubs of expertise in South Africa, predominantly in Gauteng and Durban and in the Western Cape. And so what we are wanting to do is to spread the infrastructure through the country more, to enable other researchers in other provinces in South Africa also have access to technology. If you can go to the next slide. Thanks, Tim.
Oh, I don't seem to have control. Let me try this. Okay, there we go.
Okay, great. Thank you. So a little bit more about our Nanopore working group. So we set this up at the beginning of this year, and currently there are 8 labs in our network who elected to be part of this initial phase. And what Diplomics did is that we supplied each lab with a Minine Mark 1C starter pack. So this is a very low cost starter pack. You can either get a basic starter pack or enhanced starter pack with 6 or 12 flow cells. And the idea behind this was just to give each lab a chance to test some of their DNA samples that they had in their lab, some of the workflows that they were particularly interested in their lab, just to get going with this and to compare it to other sequences that they might have generated previously. We also provide bioinformatics support through our Clarity Bioinformatics Marketplace, which Padamoya is going to talk a little bit more about shortly. And one feature of our working group is that we meet once a month in order to discuss our experiences. This comes right from ordering the different starter packs, the delivery of the starter packs. Sometimes there are different experiences that people have around that. And then also sharing tips and tricks around DNA extraction and library prep and so on. So out of the 8 different labs in our network, 2 of the labs are working on COVID sequencing. And so one of them in particular is looking at wastewater surveillance. And so those starter packs were that they purchased for their lab were the COVID sequencing starter packs. 2 labs elected to do shotgun metagenomic sequencing, one in soil and one in aquaculture samples. One of the labs was interested more in a targeted metagenomics approach, specifically 16S and ITS. And this lab is looking at identifying contaminants during the malting of barley during the beer production process. And then the last workflow that is under evaluation is whole genome sequencing, and this is obviously relevant to AfricaBP Genome. One of the labs is looking at an indigenous sheep species found in South Africa. And these slides actually here, these photos here in the slide are an illustration of the first Minine run that was done. This is at the University of Stellenbosch. And that picture in the middle there just illustrates the Mark 1C. It's really small and tiny, and that shows its suitability for this kind of research. You can pick that up and move it to the next door lab. You can pick it up and move it across the country if you want to as well. And another lab in our network is also looking at sequencing bacterial isolates. So I included this here just to show that we're a diverse group. We have lots of different interests, but so far, by all accounts, the nanopore sequencing is holding up, and people are getting interesting results from it. Tim, could you go to the next slide? Thanks. This is the last slide I've got to talk about, and it's really just to illustrate one of the new high impact projects that we're getting involved in. And this is on an indigenous plant, and it's being spearheaded by one of our partner labs, Sengen. As Tim mentioned, they're a small plant genomics lab that's based here in the Western Cape. So the plant is speckboom, or also known as pork bush. The scientific name is Portulacaria afra, and it's found extensively in the Eastern Cape province of South Africa. It's an indigenous succulent with CAM metabolism, which means that it's able to close its stomata during the day to prevent water loss. And then at nighttime, it opens the stomata and it allows for carbon dioxide to be taken up then. It is palatable, so it has feed potential for both wildlife and also for domestic animals. And increasingly, it's also become a very popular ornamental plant. So this is a pilot project to use Oxford nanopore sequencing technology to sequence speckworm here in South Africa. And the project is started just last week, and it has 3 main aims. The first of these is to develop a high molecular weight DNA extraction method. As you can imagine, out of a succulent, there are unique challenges to extracting DNA out of this plant. And then through the network, the idea is to then use 3 MinION Mark 1Cs to do a pilot sequencing project. So with a starter pack of 12 different flow cells, Sengen is going to see how far they can get with this. They've got access to 3 different instruments, and the idea is that they'll just run those instruments until they generate sufficient sequencing data. And then in collaboration with collaborators, both in South Africa and also at the John Innes Center, the idea is to then test and implement different genome assembly pipelines. So we're very excited about this project, and we hope that this is going to be the first of similar genome sequencing projects in South Africa. With that, I'm going to hand over to Padamoyo.
Thanks, Shane. So I'm Padamoyo. I'm the Bioinformatics coordinator at Diplomics. Then I'm going to talk a little bit to end the presentation, talk a little bit about bioinformatics. So as Tim mentioned earlier, each of the subgroups of Diplomics, so proteomics, genomics, and bioinformatics is made up of different hubs. So bioinformatics is a bit different because our different labs form a scientific advisory committee. So this helps inform decisions which are key to successful initiatives. So you'll see one of the pillars of Diplomics is information and intelligence. So we use these group of experts as a sounding board or as a source of information that help us best shape our initiatives. So with that, we have at this stage seen 4 main needs or areas of interest, which are to establish bioinformatics as a service, to improve bioinformatics at project design, enable or improve bioinformatics training, and inspire the next generation. So the 4th one Tim was already touched on, where you spoke about the outreaches to high school. I'm going to speak about the first 3, mainly to touch on points that align with the AfricaBP Genome project. Next slide, please, Tim. Okay, so bioinformatics is a service. So we have Clarity, which is our bioinformatics marketplace. In there, we host an expert set of bioinformaticians who are managed by a concierge. So that's just a screenshot from the website. So if you encourage you to go on the website and check out Clarity, the marketplace. It's virtual, so it's accessible across the globe. It's currently subsidized for researchers to help establish it as a service, or also show its worth as a service, because often you'll find that people consider bioinformatics as an afterthought, and you think of getting either a postdoc or a PhD student to do the bioinformatics part of your project, but you wouldn't have designed your project in order to analyze it in the way that you want. So that's why we speak quite a lot, or we're quite vocal about including bioinformatics at project design. So most of the projects that we've gotten now in the marketplace are projects after the data has been produced, and they've tried to take a look at it, and they've figured out that, you know, they're quite out of their depth. So they're trying to figure out how to get the best meaning out of their data. So we're always happy to take on those projects, but at the same time, it also helps solidify the message that, you know, do include bioinformatics at the beginning of project design. So we also get involved in high impact projects like the one Shane has spoken about now. So currently with the nanopore group, one of the groups now has data, and we've started looking at it. So one of our consultants in the marketplace, Etienne, has done the analysis for that. Next slide, please, Tim. So bioinformatics at project design. So 46% of projects fail because of poor project design. Why? Because you didn't properly consider the different needs for data analysis. So for example, if you want to count how many apples are in a basket, you need to collect apples, you know. So sometimes people want to compare 2 treatment groups and they only process 1. It sounds like such a simple thing, but we have found that these things happen. So efforts by Diplomics have been to create awareness and create an offering, which is Clarity. So create awareness is to run campaigns like this one here on the left, where we just put out very light-hearted pictures. So Patrick Shores do the right thing the first time. So this will catch your attention, but it will also has a little bit more deeper meaning that, you know, when you want to now start considering bioinformatics at the end, it will really be patchwork sometimes. And then the other one is a project without bioinformatics. It's like a car without wheels, so it's not going anywhere. So your project would have spent lots of money designing a project, running the sample, sending them wherever, it being processed in the lab, and then get data that doesn't help you answer your research question or answer your objectives. So, you know, it's like a car without wheels, it's not going anywhere. And then on the right, there is just a pamphlet for Diplomics Clarity, the marketplace, where we basically take you through if you come in and you have a project, like how we would evaluate it and the process up until you get a result. Next slide, please, Tim. Okay, so bioinformatics training. So we do it mainly through partners. As Tim mentioned in the beginning, we're new and we're quite a small group. And we also are not trying to reinvent the wheel. So if there's someone who does training quite well and they're established and they also know the market, they also know what's currently happening, we would prefer to partner with them to reach our researchers or whoever we're collaborating with. So on the right, there's just 2 examples of bioinformatics training, and we've collaborated in different ways. So for the first one is with Thermo Fisher, which Tim has already mentioned, who is a partner. And then the bottom one there is ICGEB. They're not necessarily a partner, but we align with the topic that they're focusing on. So it's omics datasets for biological insight. And we know, for example, that that would assist or that would help further projects for labs that are in our network. So we would then collaborate with them to host it. Okay, so we have 2 main groups that we offer bioinformatics training to. So the bioinformaticians themselves, in order either to upskill or to develop a new skill, and wet lab scientists, which we have found that it's quite important. It helps bridge the gap between them and the bioinformaticians. So it's not necessarily that they're going to do a bioinformatics course and they'll now know how to code, but it'll be an opportunity for them to know, for example, how to then communicate with a bioinformatician, how to communicate certain needs to get the best out of the project. So with both groups, it helps better refine the project design at different levels and different stages. So training can also be tailor-made. So for now, we've just begun on the nanopore project. We envision that once we've built pipelines that work, we could either start offering it as a training in future. So for example, if the AfricaBP Genome project were to use nanopore technology in some of their projects and we already have established pipelines, we would get, for example, bioinformaticians on Clarity, our marketplace, to sort of give out training based on the pipelines that they've built. So we would be insourcing our own bioinformaticians to help sort of spread use of pipelines that we've already created. Next slide, please. Okay, and that's basically it in terms of bioinformatics. So this is all quite new for us. We're excited to be here to learn a little bit more, to see where we can align with everyone. I hope that gave you a better snapshot of who Diplomics is. Thank you.
Thank you, Paramoyo, and thank you, Shane. And that concludes our presentation.
Yeah, thank you very much, Tim, Shane, and Paramoyo for your presentation. Awesome. So
should I stop sharing my screen?
Yeah, please stop sharing this. Yes, this is time for the panel discussion where we are going to, the participants will be asking questions and then you will address the questions. So I believe that Sujai is on the call, Nigi is on the call, Tim, Shane, and Paramoyo are all on the call. So yeah, let's start to ask our question. Yes, we have a hand raised, and yeah, please unmute yourself. And
yeah, so yeah, thanks everyone. It's been a very informative session. I think this question goes to Diplomics, Tim, and Paramoyo. It would be nice to see, because I can say that majority of what you guys do as well kind of align with the AfricaBP, especially with the Open Institutes. You know, I can see some effort in low costs, things, nanopore and the rest, and the kind of training. So how do we integrate effort? Because it would be nice to, we would like to have you guys on board fully. Basically, I mean, how can this happen?
I'll start with that. So, well, thanks for that. We've actually signed a memorandum of agreement with AfricaBP Genome. So I hope that means we are seen as a partner or participant, however that's described. By mandate, our program is focused on, creating this omics infrastructure and providing support for students and researchers in South Africa. Yet, nevertheless, we recognize that we're part of a Southern African system. And I think what we could do as a start is just to ensure, for example, we're not pursuing redundant projects. So maybe it would be a form of information sharing. So we can talk to you about the species that we're sequencing. We're very early in that, so actually we would probably borrow more from you than the other way around to get an idea of, if there are species that you're already working on, then maybe that's something we could complement with work on other species. Another way to get involved, I think, is training. So we are very close to having a training lab set up where we've built a training lab, and we're going to be able to live stream and also record training. So that's something we could open up and make accessible to the members of the network. And I would also be happy to work with other people that provide training and help to facilitate access to that training. Yeah. So those are two things off the top of my head, but I'll let Padamoyo and Shane perhaps chime in if they would like to.
Yeah, so Tim, I think we're on the same page there, definitely training. And I think as we build, we want to make it reproducible. So it would be great to also align to what Africa Biogenome Project is doing. So for example, I think in our last conversations, you were talking about possibly using Galaxy. So we could, for example, now start building in the same way. So that this can be reproduced elsewhere, for example, if other labs end up using nanopore technology.
Yeah, so I think that is really interesting. I have like 3 or 4 points. I'm not sure how far you guys are with the nanopore system. I think this is an area that would like to have an immediate exploration, potentially, basically. And in terms of species, there are about 100 species that are being tracked now within the AfricaBP, and tomorrow there will also be an announcement on some species, again, like an advertisement for a particular challenge. So again, I think it would want to work very closely. Then thirdly, you mentioned about trainings and things like that, especially in South Africa. It would be nice to see how we can jointly do this, because there's a lot I think that aligns. So it would be nice to see how we can jointly do that. We are doing the same with Sujai and the Welcome Connecting Science as well for the Future program. So we want to be able to work together and jointly organize the trainings and things like that. I think that would really. So can we have? Is this something we could potentially have as well on, and maybe as an action or wherever, however I want to do this, right?
I'm happy to action it.
Right, absolutely. Okay, and maybe we can also take the conversation out in terms of exploring other species, because there's quite a lot already going on at the species level. And it would be nice to see that we don't duplicate efforts, duplicate species sequencing and things like that, yeah.
If I can maybe just make a point, we're very keen to explore using the nanopore, and certainly our first pilot project is on indigenous plants, and we really want to do more of that. But we are aware of the fact that we will want to combine that with other sequencing technology to get a full genome sequence, maybe with Illumina or MGI sequencing in future, and maybe that's also where we can help build pipelines for improving on genome annotations in future.
Absolutely, absolutely. Even I think that the approach has also been incorporating both long read and short read within the project, and so far that is what is being done basically.
In fact, I just want to emphasize, when we launched the nanopore sort of pilot study to see and try out the technology, we brought in an expert bioinformatician right up at the front. So he surveyed the different labs to find out the kinds of projects they would want to pursue, and then evaluated the available pipelines, and then where they weren't sufficient, he has developed new pipelines. So all of this, because we're funding that, would be available, and we would just need to figure out how to make that open and available. So maybe some of the experts from the European systems here could guide us on making it open and accessible, but also, protected as necessary.
So how about we have a three-way thing? Again, we can take this conversation out. I can see Sujai is proposing. Sujai is at the Sangha Institute, and then we could have it like a triangular thing, basically, and we could potentially again have it at a sub-module within the AfricaBP program or the Open Institute. So Taiwo, is this something you want to jot down as a takeaway from this workshop as well?
Yeah, definitely.
Yeah.
We're making things.
Absolutely. Yeah.
Yeah.
Okay.
Okay. We would, having addressed that concern, we'll move to the next question. Jabin, you raised your hand. Can you unmute yourself and ask your question?
Hello, sir.
Please ask your question.
Yes,
I
sir.
can hear you.
So
Yeah.
my question is that I'm working in a single-celled algae that is known as the coccolithophore, sir. So we are actually receiving a very large amount of genomic and transcriptomic data from this coccolithophores, that is algal, microalgae, but I need to assemble them into genomes and transcriptomes, sir. So I need to know this technique or the softwares. How do I can do the assembly of those data into genomes and transcriptomes? Again, sir, I had to do the analysis of these data for the quantitative transcriptomic analysis and comparative genomics. So can you please give me a brief idea about it, how to proceed with this experiment and work?
Yeah, so Jai, let's Jai.
There's no short answer, May. I think, Jabin, I think I wrote that earlier also. But here's my tip for you and for everybody else. There are courses out there on Genome Assembly. Search for tutorials online on GitHub and elsewhere. And lots of people have offered them as courses and as tutorials. You'll have to make that effort yourself because that's not something we can answer quickly. All we can do is point you in the right direction. I hope that helps you. It's too big a topic. It's not something I can just tell you, do this software, this software, this software. There are many, many tools out there. It depends on your data. There are many tutorials out there. You'll have to find them. One recommendation that I do have for Taiwo and AfricaBP Pilot and everybody is maybe you can create resources for the community, either as a website or something saying, these are the best tutorials we found online, and that might help people like me, Jabin.
Yeah, absolutely. Thank you, Sujay. That's a good suggestion.
Okay,
I mean,
sir.
Taiwo, that is again something that another takeaway from this as well, whereby we could have on the website. So I think all of this has to do with the, as with just institutionalized AfricaBP and with the potential announcements over the next few weeks. This is something we need to take on board. So Taiwo, if you again note it down, that would really help, so that when we do the review, then we could capture that and have that populated on the website, because we get this email and suggestion and question all the time, and our aim in AfricaBP is to be able to reach out to people who would otherwise not have access to these things, and sometimes can be confusing with so many software and things, really.
Exactly.
So, yeah, absolutely.
Definitely, things are noted. Thank God. Yeah, so we ask the next question. We are going to ask Sunday to unmute and then ask his question. Sunday, please go ahead and ask a question.
Okay, my name is Obaje Godwin Sunday from the Department of Anatomy, Alex Ikweme Federal University, Nigeria, that have attended this wonderful conference on genomics. Thank you very much. Now my question, or an observation. Please, before then, please, sir, my school, my institution will be glad to have a center of this genomics in the university. Is there any way I can get a copy of your proposal that I could submit to the institution so that we, the younger lecturers, especially my area of specialization is biological anthropology, where I want to use conversation genomics to present phylogenetic trees along ethnicity and regionalization. Hello, sir.
Yeah, so thank you, Sunday. So maybe I would take this, basically, if all the speakers, because this is more to the AfricaBP framework. So what I would suggest is we're going to take this out of here. So you send an email to Taiwo. So you have Taiwo's email, right, Sunday? He has been communicating with the participant. So you send an email to him. Taiwo will then send it to the AfricaBP chairs or to myself. And then we will send you a document you will fill out to have your institution come on board AfricaBP. And yes, I agree with you. The aim of this project is to target institutions on the continent who would otherwise not have the opportunity to have access to this technologies and this knowledge and this capacity. And I think that is what AfricaBP is all about. And we've been working towards that. So you just do that, and then someone will get in touch with you, and then you can carry on conversation from there. Is that okay?
Yes, sir. I'm grateful. Thank you, sir.
You're welcome. Yeah.
Yeah. Yes, we still have more questions, but this time we're taking these questions from the chat bars.
I can answer the first one because it was to do with, So I'm going to read out the question. I think it was from Abdullah saying how, Abdullah Sharaf, whom I know from other places. The question was, how do you tell the difference between a contaminant and an endosymbiont? And I found one example. It wasn't there in my examples that I gave you today. I'm just going to share my screen quickly. This is a new example that I've added on my slide 18. So I had only these 3 examples earlier. I've now added a mollusk, Elysia chlorotica. And as far as I can see, it has a proteobacteria, either contaminant or cobiont, and it has what I think is an endosymbiont. And here's how I will show you that. So this is the genome assembly for that. It's a very messy genome assembly. This is again a public genome assembly. It's available on INSDC. You can see all the different things it has. But notice all the different colors are in one blob over here. So even though it's got chordata, mollusca, other things, they're all centered in the same location. So it's likely to just be slightly different taxonomic annotations, but it's probably from the same organism. Now, notice this big red thing that's separate as a blob. That's proteobacteria over here. If I go to filters and I go down to where it says best sum order phylum, I can disable everything. So toggle all the categories off. It actually takes more time to do that. I'm just going to enable that. Disable proteobacteria. So it's made the red blob disappear. And this button does an inversion. So it unselects all the ones that are selected and it selects the one that's unselected. So I click on that. And now only the proteobacteria is alive, visible. So you can see how there's 2 blobs, one over here, one over here. And if I now show you the mollusk itself, you can see that the proteobacteria number 1 up here is separate. So it has a different coverage. So if this is 10, that's closer to about 50 or 60. We can zoom in if you want. And this one has exactly the same coverage and the same GC as the main mollusk blob, the target organism. So my guess would be that this proteobacteria is an endosymbiont. I can't be sure of it. And this proteobacteria is either a contaminant or a cobiont. That means it was present in the sample, but it's not an endosymbiont. Endosymbionts are bacteria that are present inside the cell of the target organism. And so that's why they have the same coverage and the same GC, because they're using the same, what are they called? Codons that are available to the cell. So this would be my guess. And this is an example. I've put the link in here. I hope that answers Abdullah's question. Should I go ahead with the second question that was for me?
Yes, please.
Okay. The other question was, The other question was, how do you tell the difference between the tree and the cobiont, the proteobacteria, in the example that I showed you? So I had said it, and if you go back and look at the slides or look at the video, you might see it, but I'll say it again. So if you go to Casuarina equisetifolia, got it. If you go back here, you see 2 clear blobs at different GC, different coverage. That is always a sure indicator that these are different species. And the green color up here says Streptophyta, and the blue color over here says Ascomycota. So that's how you know that the Streptophyta is the tree, because that's the phylum of the plant, of the tree. And Ascomycota is the phylum of the fungus, which is infecting it. If you want, you can go in and identify the exact fungus, which I showed you earlier by going to filters, by going to table, by going down to best sum order genus. And then you see the Streptophyta is possibly these genuses, and I'll just do it the other way around. It's too long a list. I'll have to show it to you slightly differently, but I'll stop because this is the question answer session. But yeah, those two blobs are the two different contaminants.
Yes. Thank you very much, Sujai. We are about to round up now, but we have some other questions I would like Tim and Shane and Peter Moya to address. So the question is, it would be nice to know how you are successfully driving the linkages between diplomics and the academics, commercial and industrial laboratories in South Africa. Then there are other follow-up questions that are, what about increasing capacity building initiatives at subnational levels? And then there's this other question that, is there a need to look into how to rapidly increase skills and know-how in every aspect of the genomics process? So these are questions that I think are directly based on your presentation, if you have answers to them to address.
I'll take the first one, and then I'll let Pademoyo and Shane talk about the training and the genomics work. So the first question is actually quite good. How do we bridge across these different groups? So we are primarily tasked with supporting the academic community, and that's kind of broadly defined. We also include science councils and other similar research bodies. And the idea is, as a research infrastructure, we are an enabler. So we help enable researchers to do their work. But we're not exclusively in an academic zone. And one of the reasons for that is we also support what is called the bioeconomy strategy. This is a major governmental policy on exploiting the value that comes from bioeconomy activities. And that would include bioconservation and biodiversity studies, but also supporting things like the agricultural industry. So in that example, we do cross over with industry representative bodies. This is particularly the case in agriculture with barley and wheat growing societies and also forestry groups where they are conducting omics research. And in a commercial space, it's, outside of companies like Nkaba, there aren't many pure commercial businesses in omics. There are a few. But they tend to focus on high throughput clinical type environments or other similar clients. So you could almost say that an answer to this is, who are these labs serving? But ultimately, we see them as all being linked. So in other words, through training and through skills development, we improve the quality of omics research in the country, and then eventually, students or people in those academic groups move on and get jobs in professional companies, and that's all connected and meant to be a downstream impact from what we do. How we do it is very complicated. So, there's also a psychological element involved. The academic community is often very well embedded. What I have noticed, what we've noticed, is incredible lack of awareness of what other people are doing, even in the same departments or in the same subject areas. So this speaks to our visibility and awareness objectives. So all of these are meant to dovetail and intertwine to create an overall a stronger omics community and an omics space in South Africa. I hope that answers your question.
Yeah, definitely. I think the answer has been provided. Thank you so much, Tim.
Can you remind us of the other 2 points, please?
Yes. The other question is, what about increasing capacity building initiatives at subnational levels? And then the other one is, there is a need to look into how to rapidly increase skills and technical know-how in every aspect of the genomics process. So just your take on this.
Pademoyo, would you be willing to tackle the first one about skills development at subnational level?
Sure. So maybe, and also a little bit about the second one. So, and just touch on the first one when we're talking about commercial. So I think another aspect of diplomics, the information gathering one, is sometimes the skill that you want to build is for a season. So for example, it's a master's student who needs a skill for their project, and then they're going to move on. It might not necessarily add value to their project for them to be the hands-on person who's using that skill. So that's how sort of Decipher and Clarity as a commercial entity in Diplomics comes out, right? So you would then send someone to do the service. So doing bioinformatics is not necessarily a core part of your project or your research project, so you outsource that. So all of these things, I think for us, when we make decisions, that's why we have a group of experts that form a scientific advisory committee who would help us inform certain decisions that we make. So for example, if we choose to rather have a bioinformatics marketplace than to upskill bioinformaticians, it might be because the information that we're getting is for 90% of our students, bioinformatics is not a core part, but they have data that they need to analyze. So I think for each decision or path that we decide to take, it's informed by our partner labs, what we're currently experiencing in South Africa. It's very contextual. So I hope that that answers the question.
Yeah, definitely. Thank you so much. But to finalize this section and then to close the workshop for the day, we have one last question. So Nigi, how is Nigi and Kate? How is the Uniprot database increasing data for plant and animals?
Okay, fine. Yeah, so most of our sequences actually come from other databases. They're translations of nucleotides, so they're from ENA, DDGB, and NCBI. So we're expecting that as the cost of sequencing comes down and more organisms are being sequenced, they'll be going into ENA and Ensemble and DDGB and NCBI, and then we'll be pulling them into our database. So that's how we're expecting. If the data is there, we pull it in to Uniprot. So as sequencing and data increases, the coverage of species will increase in our database as well.
Yeah, thank you so much, Nigi and Kate, for providing answer to that question. At this point, we have come to the end of this section, and want to, on behalf of the organizing committee and the AfricaBP project, then thank all our facilitators, and especially the facilitators for today, Sujai, Nigi, Tim, Shane, and Peter Moya, for your time on this section. And to all our participants, we are thankful for your presence, and we hope that you have learned so much from this workshop. And then we'll get back to all participants with respect to resource materials and all other things that will be given out to participants. On this note, I'd like to, on behalf of the organizing committee and the AfricaBP, thank you all for your participation. Then say bye to everyone.
Thank you, everyone.
Thank
Bye-bye.
you. Bye.
Great.